Dobro jutro. Dobro jutro in lepo pozdravljeni. Moje ime je Robert Zorec, jaz bom danes povezoval in bom najprej seveda prosil podpredsednico Slovenske akademije znanosti in umetnosti, akademikinjo Tatjano Avšič-Županc, da spregovori nekaj besed o tem dogodku. Tatjana.
Spoštovani, en lep dober dan ali dobro jutro. Torej v imenu Slovenske akademije znanosti in umetnosti vas vse današnje predavatelje in pa seveda tudi cenjene udeležence srečanja Translacijska medicina, Nevrologija prisrčno pozdravljam. Zdaj bom nadaljevala ta pozdrav v angleščini, predvsem zaradi naših predavateljev in cenjenih gostov iz tujine. On behalf of the Slovenian Academy of Sciences and Arts, I would like to welcome all the speakers and all the participants, especially the professors of secondary schools. There are two main reasons for today's event. First, the Slovenian Academy of Sciences and Arts has been in continuous existence since November 12, 1938, when the original group of Academy members met for their first session. Therefore, this year, we are celebrating an 85th anniversary. But more than 300 years ago, exactly in 1693, the forerunner of today's Academy of Sciences and Arts, so-called Academia Operosorum, was founded in Ljubljana. This means that our academy, which dates back to the end of the 17th century, devotes special attention particularly to the promotion and advancement of science through symposia and workshops like we have today. A similar meeting on the other brain, it was held here in 2019. In September next year, that means 2024, we will also co-organize the International Conference on Brain Energy Metabolism, which will focus on topics related to basic research and translation. It is worth also mentioning that today's workshop also promotes the recently signed international cooperation between the Zhejiang Chinese Medical University and the Faculty of Medicine of University of Ljubljana. The topics of this collaboration will be today presented, and we welcome very much Professor Vladimir Parpura, who is also the member of our Academy of Sciences and Arts, Professor Montana, both from the Zhejiang Chinese Medical University. It is also a special privilege to welcome Professor Alessandro Marcello from the International Center for Genetic Engineering and Biotechnology from Trieste, who is collaborating with us on virus research. At the end, we would like to collaborate also with Professor Dimitri Krainz from Chicago, who will be the last speaker of the day, actually online. But not the least, we also are very, very proud that we will hear our younger colleagues presenting their recent and unpublished research today. What is the second reason? The second reason is that the Nobel Prize Award ceremony in physiology or medicine is not far off. We have dedicated the first lecture, therefore, to the discoveries of the 2023 Nobel Prize winner, and we are grateful to Professor Alois Ihan, my dear colleague, for agreeing to giving us a lecture on this occasion. I would like to welcome you once again and wish you all the productive and enjoyable work at our academy, which still identify itself the mission with the statement, sis apis, which means be wise as the hard-working bees. Thank you very much.
Many thanks for your address, and I'd like to invite now Professor Nina Vardjan, the President of Slovenian Academy of Sciences and the President of Slovenian Physiological Society, and Professor Margo Kreft to continue.
Pozdravljeni v imenu Slovenskega fiziološkega društva, cenjeni gostje. Danes bo potekal dan fiziologije v okviru delavnice Translational medicine Nevroglija, torej v soorganizaciji s SAZU, Univerze v Ljubljani medicinsko fakulteto in pa Slovenskim fiziološkim društvom. Letošnja Nobelova nagrada je bila podeljena za odkritja v zvezi z modificiranjem baz, ki so omogočila razvoj učinkovitih messenger RNA oziroma RNK cepiv proti COVID-19. Dan fiziologije, kot veste, vsako leto poteka na temo Nobelove nagrade, in to leto sta jo prejela dva znanstvenika, ki sta prispevala zelo pomembno k razvoju cepiv proti COVID-u, in sicer Katalin Kariko in Drew Weissman. Več o tem boste slišali v našem naslednjem predavanju. Imamo gosta, profesor dr. Alojza Ihana, ki nam bo povedal in imel naše osrednje predavanje danes v zvezi z Nobelovo nagrado. Pri tem bi rada povabila profesor Krefta, ki bo predstavil profesorja. Hvala lepa.
Naš prvi govorec bo profesor dr. Alojz Ihan, redni profesor za imunologijo in mikrobiologijo na medicinski fakulteti Univerze v Ljubljani, je zdravnik specialist za mikrobiologijo in za klinično mikrobiologijo, je predstojnik katedre za mikrobiologijo in imunologijo, poleg tega tudi vodja oddelka za imunologijo na Inštitutu za mikrobiologijo in imunologijo medicinske fakultete Univerze v Ljubljani. Za svoje literarno delo je prejel nagrado Prešernovega sklada in za svoje udejstvovanje, povezano s temo današnjega predavanja, je prejel leta 1921 nagrado Komunikator znanosti. Lepo prosim profesor Ihana, da predstavi to tematiko.
Ja, hvala lepa za povabilo, da bom v tej luštni dvorani, ki je ravno prav velika, pa ravno prav mala, da človek z veseljem predava. Torej, tokrat seveda se bom opisoval tole zgodbo teh dveh odkriteljev, se pravi Katalin Kariko in pa Drewa Weissmana, ki sta prispevala, kot bomo videli, en delček, ampak res pomemben in pa predvsem aktualen delček, ki je v veliki meri omogoča razrešiti epidemijo COVID-19. In ob tem bomo seveda, moj namen je predvsem, da malo pokažem še druge stopinje, ki so vodile do tega cepiva. Boste videli, da jih je zelo, zelo veliko. In pa seveda začel bom s tem diasom, ki, jaz sem še generacija, ki je imela diase, težko se z drsnicami spopadam. To je pa roman oziroma gotov velik filmov zaradi zgodbe, se pravi z Drewom, se pravi z Weissmanom se najbrž ne bodo filmi toliko ukvarjali kot z odkriteljico, zaradi njene zgodbe, se pravi, ona se je začela že leta 1978, to je bila pravzaprav njen doktorat na Madžarskem, se je začela ukvarjati z mRNK in sicer s tem, kako bi z mRNK vplivala na celice, da bi tisto informacijo, ki je v mRNK, da bi tisto informacijo enostavno prenesla v celice in da bi celice ubogale, se pravi, da bi delale proteine, kot se jih inicira v celico. Se pravi, na Madžarskem je potem nekako, bom rekel, poklicno prišla v pad pozicijo, to bo dober film spet, verjeten, lahko razmišljate, katera igralka bi bila primerna. No in potem je emigrirala v Združene države, na Pensilvanijo in na Pensilvaniji pravzaprav bi težko rekli, da je bila zelo uspešna raziskovalka. Torej, kar s težavo se je prebijala skozi projekte kot večina raziskovalcev, dokler ni prišla v laboratorij Drewa Weissmana. Pri njemu je, skupaj sta potem v reviji Immunity, ki ni ravno neka top revija, sta objavila način, kako se z mRNK reprogramira celice. No in potem je zaradi tega načina, ta članek, ki ga bomo pol videli, je zanimiv po svoje, ker ima neverjetno razsežnost. Po eni strani dokaže fenomen, po drugi strani že najde tehnološko rešitev, da se ta fenomen bypassira. Torej, redko kje najdemo članek, ki bi tako na široko, pravzaprav ne samo, da nekaj odkriješ, ampak da v istem članku že kar poveš, kako boš to težavo, ki je nastala z mRNK, ki jo damo celicam, kako boš to težavo rešil. Zdaj, glavna težava je bila seveda v tem, da je mRNK, ki jo damo celicam, da je hudo vnetna. Torej, mRNK je nekaj takega kot adjuvans, ko ga damo celicam. Se pravi, se prime na receptorje, tolične receptorje in povzroči vnetje in seveda dajati mRNK, vbrizgati v žival ali pa človeka ni pomenil, da se dirigira celice, ampak je pomenil, da se naredi SIRS, se pravi vročina in pa potem vsi znaki sistemskega vnetja. In zaradi tega, in ker je ona to, ker sta z Weissmanom objavila vzrok, zakaj mRNK povzroči vnetje, sta v istem članku potem tudi pokazala, kaj narediti za mRNK, da ni tega vnetja. No in ker je bil ta recept toliko unikaten, je seveda takoj sledil naslednje leto patent in po patentu, in ta patent, ki je bil univerzitetni patent, ker je pač delal v univerzitetnem laboratoriju, se pravi, to je bil od Pensilvanije patent, je tale patent potem kupil zelo pomemben molekularni biolog tistega časa, Derek Rossi, in z tem patentom je ustanovil potem podjetje Moderna. Zaradi tega, ker je nekako mu je postal jasen, da če lahko z mRNK naredimo ekspresijo v celici in vivo, je potem pravzaprav to način za delati zelo čisto cepivo. Kaj pomeni čisto cepivo? Ne tako, kjer patogena speštamo, pa vse skupaj vbrizgamo v človeka, ampak tako, kjer samo tisti ključen protein izrazimo, brez vseh drugih primesi. In to je seveda Rossija tako navdušilo, da se mu je zdelo to zelo perspektivno, da je ta patent odkupil. No in Katalinkari, ko je delala potem seveda v kombinaciji za Pensilvanijo in pa za Moderno tudi, vendar nekako nista imela velike kemije z šefom Moderne, se pravi z Rosijem, tako da je pravzaprav dala odpoved pri Moderni in potem je se nekako dogovorila pri nemškemu biotehnološkemu podjetju BioNTech, ki sta ga vodila, ki ga je vodil Sahim pa Tirecijeva zakonski par in je bil ustanovljen leta 2008 in nekako bolj zaradi te prezaposlitve je potem leta 2013 Kariko naredila še en patent, ki je omogočil z malo drugačne strani to modifikacijo, ki jo bomo pol videli. In to je bilo pravzaprav, to je bila njena zgodba, za katero je malo kdo vedel, dokler ni prišel COVID. No, zdaj o cepljenju ne bom dosti govoril, čeprav se ga bomo skozi nekako lotevali. Se pravi, cepljenje vemo, da je način, kako posnemamo okužbo, da postanemo odporni. Se pravi, po nekaterih okužbah so že zapiski iz stare Grčije, da če prebolimo neko bolezen, jo potem več ne dobimo. In cepljenje je samo način, da na čim bolj, bom rekel, naraven način skušamo preboleti prvo okužbo, s tem, da nimamo posledic bolezni. No in pri nekaterih in pri cepljenju pravzaprav povzročimo isto, kar se dogaja pri okužbi, torej razmnožimo limfocite. Limfociti za razmnoževanje rabijo vsaj en teden, da se namnožijo iz enega v 5 do 10 tisoč, to je tista količina limfocitov, ki naredi dosti protiteles, da nas zaščiti pred okužbo. Se pravi, rabimo vsaj en teden razmnoževanja, pa še nekaj priprav, tako da nekako v 14 dneh do enega meseca smo nekako s cepljenjem na istem, kot če bi preboleli bolezen. In torej pri nekaterih boleznih se to res plača, to je zlasti pri tistih boleznih, pri katerih imamo pojav, da zbolimo samo enkrat, potem pa ne več. Pri večini okužb se pa žal to ne splača, zato ker če je okužba taka, da poteka kronično ali pa večkrat, navadno tudi cepljenje nima kakšnega večjega pomena, ker nas torej cepljenje nič drugega ne naredi, kot samo povzroči efekt prve okužbe. Torej, če se neka bolezen kronično ponavlja, ni kakšne velike koristi od tega, da naredimo prvo okužbo, ker se bo itak potem enako ponavljala. No, se pravi, cepivo pravzaprav posnema okužbo in mora vsebovati dve bistveni sestavini vsake okužbe. Eno so tisti ključni proteini, proti katerim bi radi naredili imunski odziv, te imenujemo antigeni. Drugo so pa snovi v steni mikroorganizmov, ki pa najprej vzdražijo makrofage, da začno požirati mikroorganizme in taki jezni makrofagi potem odnesejo vse skupaj v bezgavke, kjer se naredi imunski odziv. Oboje rabimo pri cepivu. Se pravi, pri okužbi je eno in drugo prisotno. Je stena mikrobov prisotna, ki vzdraži makrofage, in pa seveda je prisotna tista tarčna molekula, proti katerim se delajo protitelesa. In odgovor recimo dobimo potem protitelesa. Zdaj govorimo o klasičnih cepilnih platformah, to so tiste, ki se jih uporablja že odkar je Jenner naredil prvo cepivo, potem pa Pasteur pogruntal princip delanja tega cepiva. No, to je značilno pri raziskovanju, da pravzaprav velikokrat se najprej naredi efekt, potem pa se šele razmišlja, zakaj je ta efekt pravzaprav nastal. No in se pravi, klasična cepiva so pravzaprav v glavnem samo mikroorganizmi, ki jih na nek način zatrejo in ti mikroorganizmi imajo vse, imajo in antigen in imajo tiste aktivatorje v stenah. Modernejša cepiva so pa taka, kjer rečemo, zakaj bi celega mikroba brizgali pa naredili stotine imunskih odzivov, če pa rabimo v resnici samo en imunski odziv proti točno določenemu proteinu. Zato se ta cepiva, jih imenujemo proteinska cepiva, torej vzamemo samo en protein, ampak če vzamemo samo en protein, se znajdemo potem v tisti zanki, kdo bo pa makrofage naredil jezne, da bodo to požrli. No in klasično so videli, da to krasno deluje z kovinskimi opilki, torej če vzamemo protein, pa raztopino proteina prelijemo prek kovinskih opilkov, ki bi jih danes imenovali seveda nanodelci. Potem se na teh kovinskih opilkih naredi film iz antigena in to makrofagi požrejo, zato ker so programirani, da požirajo vedno delce tudi, in vse skupaj odnesejo v bezgavke in najbolj uporabljena kovina je seveda aluminij pa njegovi oksidi. Lahko bi pa tudi druge kovine enako dobro delujejo. Moderna cepiva pa imajo namesto tega, odkar so odkriti tolični receptorji, pa uporabljajo kar agoniste toličnih receptorjev. Se pravi, makrofage se da narediti jezne na ta naraven način, s tem, da damo kar agonist toličnih receptorjev, recimo iz salmonele se dobi lipopolisaharid, aktivna snov v lipopolisaharidu je lipid A in ta lipid A iz salmonele je del številnih cepiv, ki ga vsaka tovarna seveda zaščiti z neko kratico in nekim patentom, ampak v resnici je več ali manj so to samo lipidi, sintetični lipidi A, ki so bili originalno iz salmonel ali šigel ali mešerihi, to je pač stvar tovarne posebej. Torej, makrofagi imajo tolične receptore za prepoznavo bakterij, pa tudi za prepoznavo virusov, in tudi ti agonisti, recimo toličnih receptorjev 3, 9, 8, ki jih dražijo različne oblike DNK pa RNK, tudi ti agonisti so čist v redu adjuvansi. Se pravi, lahko bi uporabili tudi RNK namesto lipopolisaharida kot dodatek k cepivu. Ampak ta RNK ne bi deloval kot, da prenaša informacijo, ampak deluje kot to, da vzdraži makrofage, naredi vnetje, da ti potem odnesejo vse skupaj noter. No in tukaj je en zanimiv članek, ki je izšel leta 2014 in tukaj vidite zgodovino tehnološkega razvoja cepiv. Začne se seveda v stari Grčiji, kjer je prvi zapisek, da ljudje, ki prebolijo kugo, so potem lahko skrbeli za bolnike, zato ker ni bilo več nevaren, da bi spet zboleli za kugo. Se pravi, samo tisti ljudje, ki so enkrat že preboleli atensko kugo, so lahko skrbeli za bolnike. Vsi ostali so zboleli, če so skrbeli za bolnike. In ta princip, da izpostavitev bolezni pomaga, se pravi, da ko prvič prebolimo okužbo pri nekaterih boleznih, ta princip so tudi kasneje včasih izkoriščali, recimo so delali zabave za otroke, za ošpicam, ko je nekdo dobil ošpice, so naredili partije za to, da so otroci zgodaj dobili ošpice, da niso kasneje dobili, ko so hujše bile. No, potem klasične platforme za izdelavo cepiv se delijo na stare, to je tisto, ko se nagoji mikrobe, se jih na nek način uniči in to cepi, in pa potem novejše proteinske, ko se uporabi z čiščenjem ali pa z ekspresijo v kvasovkah, recimo samo en protein in se ga da za cepivo. In ta članek iz leta 2014 pravzaprav se v glavnem ukvarja z perspektivo cepiv v smislu proteinske tehnologije. Se pravi, kot prihodnost so videli izključno bolj sofisticirane načine pridelave proteinov. Potem pa praktično z enakim naslovom članek iz leta 2021, tukaj pa vidite, da se stvar naenkrat zelo spremeni. Se pravi, ni veliko let vmes, ampak naenkrat se kot perspektiva pojavijo DNK in pa RNK cepiva. Se pravi, in vzrok zamenjave te perspektive je spet COVID. COVID je naredil, da se je o cepivih začelo razmišljati čisto drugače. No in COVID je povzročil, da sta nastale za humano uporabo dve novi tehnologiji. Ena tehnologija, ki sploh ni bila nova, in sicer je tehnologija vektorskih cepiv, kar pomeni namesto uporabimo en modelni virus, v njega damo gen, proti katerim želimo, da nastane protitelo, in ta modelni virus, ki ni patogen, recimo adenovirus, s tem virusom se cepi žival ali pa človeka, lahko je v obliki živega virusa ali pa virusa, ki se ne replicira, pač pa še vedno dela proteine. No in v tem, ta tehnologija je torej niti slučajno ni nova tehnologija, to se množično uporablja za živalska cepiva. Se pravi, to so veterinarska cepiva, zato ker je tehnologija izredno poceni. Torej, ko se enkrat naredi ta vektor, je razmnoževanje teh adenovirusov silno poceni in pri veterinarskih cepivih je seveda zelo važno, da cepiva niso dražja kot pa žival, ki jo želimo na koncu prodati, ne. Sploh, če gre že manjše živali, ne pride skozi. Druga tehnologija je pa tehnologija, ki je bila dejansko uvožena iz razvojnih laboratorijev, to je pa tehnologija, za katero je bila ta letošnja Nobelova nagrada, se pravi tehnologija cepljenja z mRNK. Kako se je to začelo? Se pravi, vsaka stvar ima svoj začetek in to je verjetno prvi članek iz leta 90, ki je opisal, da je mogoče iz RNK ali pa DNK, da je mogoče pripraviti injekcijo, vbrizgati v žival, v miško in pokazati ekspresijo čez nekaj dni, in ta ekspresija je trajala nekaj dni do nekaj tednov. Se pravi, to je bil verjetno eden od prelomnih člankov, mogoče si zapomnimo avtorja Malona, ki ima veliko pri tem razvoju, ko bomo pa razmišljali, kdo dobi Nobelovo, pa kdo ne. Gospod, imaš precej zamer zdaj, ravno zaradi te Nobelove, ne? Se bomo pol videli. Torej, se pravi, to je prva, se pravi, to je razvoj cepiv, mRNK cepiv do COVID-a. In ena od najpomembnejših zadev je bila seveda dokazati, da je vbrizganje mRNK, da ima sploh kakšen učinek. In to je bil ta članek iz leta 90. In potem pa pride že 2005, se pravi, pride ta članek, kjer sta oba Nobelovca zdajšnja pokazala, da je mogoče, da pravzaprav RNK draži tolične receptorje in zaradi tega deluje kot adjuvans, se pravi, povzroča vnetje. Zakaj sta ta članek lahko naredila šele leta 2005? Zaradi tega, ker so bili tolični receptorji samo malo prej odkriti. Torej, pred odkritjem toličnih receptorjev so vedeli, da mRNK povzroča vnetje, ampak ni bilo jasno zakaj. Ko so odkrili tolične receptorje, so videli, imamo posamezne, ki primejo na dvojno vijačno RNK, imamo posamezne, ki primejo na dvojno vijačno DNK, se pravi, ko so to odkrili, je bil eden od člankov, ki ga je seveda Kariko naredila, da je ugotovila, zakaj mRNK povzroči vnetje, in vnetje je dokazala, da ga povzroči zato, ker se veže na tolične receptorje 3, 8, 5 in zaradi tega povzroča draženje, makrofagi začno delati vnetne citokine, interleukin 6, tumor nekrozni faktor. Tisto, kar je pa še dodatno naredila in zaradi tega članek res zasluži občudovanje, je pa to, da je pravzaprav naredila tudi vrsto modifikacij mRNK in med njimi je tale, ki je dejansko tista, ki je v cepivih. Se pravi, to je modifikacija, kjer se namesto uridina uporablja pseudouridin, se pravi izomer uridina. To ni naravna modifikacija. Naravno so, Kariko je v tem članku pokazala, da če doda celicam človeške ali pa živalske mRNK, ne pride do vnetja. Če pa celicam doda sintetične ali pa virusne mRNK, pa pride do vnetja. Zaradi tega, ker imajo naše celice na različne načine modificirane mRNK, metilirane in tako naprej, in se ne vežejo na tolične receptorje. Se pravi, Weissman in Kariko sta pa pravzaprav testirala nekaj modifikacij, ki so čisto umetne, ki pa tudi preprečijo vezavo na tolične receptorje. In ena od modifikacij, ki se je izkazala za najbolj učinkovito, je bil ta pseudouridin. Je tudi nekaj drugih modifikacij, ki so bile nekoliko manj učinkovite in bi jih tudi verjetno lahko uporabljali za cepivo, ampak Kariko je pač našla, da je to skoraj v celoti odstrani vnetje, in to je bil potem ta ključen članek, za katerega je dejansko prišla Nobelova nagrada. Se pravi, ta članek je alfa in omega za to Nobelovo nagrado. Se pravi, odkriti, zakaj človeška mRNK ne dela vnetja, sintetična pa dela vnetje, in pa tudi hkrati pokazati, kaj narediti sintetični mRNK, da ne dela vnetja. No, potem seveda to ni bilo cepivo. Cepivo je bilo treba dati noter v celico. In tukaj je spet en prispevek že omenjenega Malouna, ki je tudi prvi na nek način odkril, da se da z kationskimi liposomi, da se da narediti mehurčke, ki vežejo potem mRNK, ko vse skupaj zmešaš, in da se s pomočjo teh mehurčkov veliko bolj učinkovito prenese mRNK v celice, kot pa bi samo dodal mRNK, recimo z injiciranjem v mišico. Se pravi, ti liposomi, ti mehurčki so bili zelo pomemben mejnik v izdelavi cepiva, in jaz moram reči, da sem osebno se srečal s tem cepivom šele tam leta 18, ko smo z profesorjem Batelinom delali en projekt na mikro-RNK, se pravi, mikro-RNK je čisto nekaj drugega kot mRNA, to je nekodirajoča regulatorna RNK, in pravzaprav smo od diabetičnih pacientov za tipom 1 diabetesa smo iz krvi izolirali njihove mRNK-je, njihove mikro-RNK-je, jih primerjali z nediabetičnimi posamezniki in potem tiste različne mikro-RNK-je smo sekvencirali, jih nasintetizirali, oziroma jih dali nasintetizirati, to so njihove strukture, in potem se je pojavila potreba, da s tem vplivamo na citotoksične celice. In zato je bilo najbolj enostavno dati se skupaj v mehurčke in jih izpostaviti citotoksičnim celicam, in ti mehurčki, in o teh mehurčkih jaz prej nisem nič vedel pravzaprav. In potem sem našel en članek, ki me je res fasciniral, to je članek o mRNK cepivih, kot so bila do takrat, se pravi pred covidom. In tukaj vidite, da v tem članku zelo lepo je shema, če dodaš mRNK tako, kot je nasintetizirana, imaš pravzaprav adjuvans. Se pravi tisto, kar naredi vnetje. Če pa dodaš mRNK, ki je modificirana, ki vsebuje pseudouridin, pa daš v mehurčke, pa iz to enako mRNK potem lahko narediš ekspresijo, ki jo želiš. No in iz istega članka imamo nekaj načinov, kako se oblikuje ta mRNK, do takrat je bilo že to zelo jasno, in pa imamo zelo različne načine, kako se oblikuje te liposomske mehurčke. Se pravi, od tistega prvega Malonovega mehurčka, ki je bil zelo preprost, do cele tehnologije, kako se različne mehurčke oblikuje za različne namene. In seveda, to je mehurček, ki gre v cepivo, o tem bom še potem govoril. No in ko smo delali torej ta članek, smo takrat uporabili en kationski lipid, ki je pravzaprav najbolj preprostejših, se pravi, ti naši mehurčki so bili taki, ki nikakor ne bili dobri za cepivo. Zakaj ne? Po eni strani jih je bilo zelo lahko narediti, ker smo samo zmešali sintetizirano mRNK z mikro-RNK z lipidi, ampak ko smo gledali pod mikroskopom, smo videli sicer krasno, se pravi, lipidna ovojnica znotraj zaprte mikro-RNK-je, ampak mehurčki so bili izredno različnih oblik. Se pravi, imamo male mehurčke, imamo ogromne mehurčke, to bi bilo za cepivo neznosno, oziroma ne bi šlo skozi. Je pa šlo skozi za poskus, kjer ni nevarno, da bo nekdo dol padel, ko dobi cepivo. No in iz tega istega članka, kot vidite, do leta 2018 so tukaj že, bom rekel, razvojna podjetja, ki so se ukvarjala s temi mRNK-ji za delanje cepiv. In med njimi seveda najdemo že Moderno in pa razvojna podjetja, ki so se ukvarjala z poskusi zdravljenja raka. Se pravi, da se vzame, koncept je zelo enostaven, vzame se vse mRNK-je, ki so v rakastem tkivu, pa mRNK-je, ki so v zdravem tkivu, potem se dela sekvenciranja, potem se primerja, kaj je razlika, in tisto razliko se potem naredi ekspresijo. In tista razlika so verjetno tumorski antigeni, s katerim lahko naredimo potem imunski odziv proti tem tumorskim antigenom. No in s tem so se ukvarjala različna razvojna podjetja in med njimi tudi BioNTech, kjer je takrat delala že Kariko. No in torej, tukaj so ti trije mejniki, se pravi narediti mehurček, pokazati, da je v ta mehurček mogoče dati mRNK, in pa seveda narediti mRNK tako, da ne bo vnetni adjuvans, ampak da bo informacijska molekula. Vse drugo je bilo pa tudi treba narediti. Se pravi, brez kakršnekoli od teh stopenj ne bi bilo cepiva. Se pravi, treba je bilo odkriti DNK, treba je bilo odkriti, kako deluje RNK, treba je bilo odkriti, kako je mogoče sintetizirati, se pravi, kako je mogoče s pomočjo genske tehnologije sintetizirati nukleotide, treba je bilo odkriti, kako je mogoče RNK sintetizirati, se pravi, vse te stopnje, brez katerekoli od teh stopenj ne bi bilo cepiva. Ampak seveda, Nobelova je bila samo za eno stopnjo. Zakaj? Zaradi tega. Vsi vemo, da nagrade zlasti v medicini, medicina je praktična veda in nagrade v medicini so navadno vedno povezane z rešitvijo nekega problema, ki je res pereč. Verjetno imajo neka druga področja, kaj s tem fizika, mogoče, da so nagrade tudi za čiste intelektualne presežke, ampak v medicini so ponavadi nagrade povezane z nekimi praktičnimi rešitvami in tisto, kar je povzročil, kar je bistveno pri tej nagradi, je seveda pandemija covida. Brez te pandemije, se pravi, covid je pravzaprav nekaj, kar, se pravi, virus, ki je pravzaprav iz neke prehladne bolezni naredil kataklizmo svetovno. Na srečo je bilo nekaj podobnega prej z nekim drugim koronavirusom, se pravi, je bilo z SARS-om prej, zato je bilo tudi nekaj izkušenj v tem pogledu, se pravi, povzročitelj covida ni bil čisto nov virus, prej je bil SARS, na srečo. In o SARS-u se je marsikaj naučil, kar pomeni, da takoj, ki je še naredila pandemija, je postala struktura tega virusa zelo hitro jasna, zato ker so bile prej izkušnje na SARS-u, ki povzroča zelo podobno bolezen. In seveda tisto, kar je bilo zelo važen, razvoj politika se je zaradi velikega problema odločila za razvoj cepiv. Pravzaprav bomo verjetno nikoli ne bomo popolnoma zvedeli, zakaj so bile izbrana posamezne cepilne platforme. Najprej je kazalo, da bo izključno adenovirusna platforma, to je bilo tudi najbolj logično, ker so bili to že živalska cepiva. Verjetno pa tudi malo zaradi prestiža se je potem pokazalo, da je v Združenih državah Moderna, da je v Evropski uniji BioNTech. Takrat se je Evropska unija že ločila od Anglije in verjetno so politiki iz Evropske unije pa Združenih držav bi težko pristali, da bo vsa tehnologija pravzaprav da bodo vso tehnologijo, ker se je vedelo, kako bo to ogromno, da bodo vse pobrali Angleži, pa še odcepili so se. Tako da so bile zaradi tega izbranih več tehnologij. Ena je bila, tu so milijoni, ki so šli v prvi obroku k posameznim tovarnam. Tukaj vidite, da je precej favoriziran bil BioNTech, AstraZeneca nekaj manj. V Združenih državah je bila Moderna tista, v katero se je zlivalo ogromno denarja. In zaradi tega je logično, da so se razvila predvsem mRNK cepiva in pa nekaj manj vektorska cepiva, ki so bila, kot rečeno, najbolj logična najprej. Zdaj, precej manj so dobivale klasične cepilne platforme, ampak kot rečeno, to je bilo prvič, da so države skoraj v celoti financirale razvoje. nekako malo čuden smo gledali potem tisti, ki smo vsem sodelovali na teh konferencah, ko smo zvedeli, da pravzaprav je bilo financiranje strani držav, ampak te pravice se pravi lastnina cepiv pa ni bila, je bila še vedno v okviru tovarn, zato ker je bil en poseben financiranje na način odkupov. Se pravi, ni bilo neposredno financiran, ampak je bilo zakup, kar pomeni, tovarna še vedno ostane lastnica vsega, dobi pa posel za naprej, ki je vreden recimo 600 milijonov ali kasneje tudi več kot milijarda, ampak če ti nekaj vnaprej zakupaš, si še vedno lastnik tega, ne? Se pravi, države niso bile deležniki, ampak so naredile vnaprejšnje zakupe. Zaradi tega je bilo to tako. No in to je potem ta sestava cepiv, to imamo BioNTech, imamo Moderno, noter, kot vidite, je precej lipidov, polietilen glikol in ostali lipidi, ki omogočajo take stabilne in predvsem neverjetno enake mehurčke. Pod mikroskopom to res gleda kot teniške žogce. Se pravi, ta tehnologija nekaj povsem drugega kot neko mešanje v epruveti. No in zaradi tega so zelo hitro nastala torej ta prva cepiva. Ta prva cepiva so se izkazala kot izjemno učinkovita, pri tvorbi protiteles so bila bolj učinkovita kot pa naravne okužbe. Tisto, kar je motilo pri teh prvem valu cepiv je, da pri naravni okužbi so protitelesa razmeroma počasi padala koncentracija, medtem ko pri cepljenju je to padanje bilo precej hitrejše, zato se je vedelo, da bo treba večkrat cepiti, če bi želeli dobiti enak učinek kot po naravni okužbi. Ampak kljub temu je cepljenje brez dvoma rešilo, če je bilo smrtnost zaradi covida na svetu ta registrirana okrog 7-8 milijonov, je bilo presežnih smrti tam nekje 10 do 15 milijonov v resnici. Cepiva so rešila tam najmanj 5 do 10 milijonov življenj v teh prvih valovih. Potem pa seveda se je virus razvijal, se je začel izogibati cepivom, ampak na srečo je bilo večina ljudi že precepljenih, kajti ko se enkrat okužimo ali pa cepimo s covidom, je potem skoraj ne bomo dobili več smrtne bolezni. Zaradi tega smo opažali potem, takoj ko se je pojavil nov sev, je bila učinkovitost cepiv bistveno manjša in to recimo so naša poročila iz našega inštituta, to je zadnje poročilo, ko se je pojavil omikron, vidimo kako koloristično pravzaprav se pojavljajo posamezne podrazličice omikrona, se pravi, virus je tako izmuzljiv, da je pravzaprav praktično nemogoče narediti cepivo za točno določen sev, ki bo v resnici uporabno. No, ampak kljub temu cepivo je opravilo ogromno vlogo. Tukaj bi pa sam še pokazal nekaj, za kar bo mogoče, bodo rekli, se bi dal Nobelovo, ampak se sta jo itak že dobila. Torej, oba proizvajalca, Moderna in BioNTech, sta potem zdaj po pandemiji nadaljujeta predvsem ta program zdravljenja raka in tukaj vidimo en članek, ki je zelo odmeval junija 2023, kjer so pravzaprav naredili to proceduro, ki so jo že prej planirali, torej zdaj že v okviru klinične študije, torej da pri zdravljenju raka trebušne slinavke, da se vzame tumor, se ga iz njega izloči mRNK-je, se vzame vzorci zdravega tkiva, izloči mRNK-je, naredi sekvenciranja in potem najde mRNK-je, ki so samo v tumorju, ne pa v zdravih celicah, in potem se te mRNK-je enostavno zapakira v mehurčke in naredi na nek način industrijski postopek za personalizirano cepivo. Se pravi, postopek, ki ga lahko pravzaprav se množično dela na stotinah in stotinah pacientov, brez da bi se poglabljali vsakega, ampak v resnici je pa personalizirano cepivo za vsakega posebej. In seveda zbrali so tistega raka, ki je že 40 let, kot vidite, rak, ki je najbolj smrten in pri katerem ni nobenega napredka bilo v zadnjih 40 letih. Pri večini drugih rakov imate ogromne napredke v preživetju, ti napredki so ponavadi povezani z biološkimi zdravili in podobni. Medtem ko pri trebušni slinavki tega napredka ni in tudi smrtnost je izjemna. In zaradi tega je seveda trebušna slinavka tisti nekak model, kjer se ta mRNK cepiva zdaj klinično preizkuša. In trenutno je to cepivo, ker je prva faza v resnici pokazala krasne učinke, čeprav je prva faza namenjena samo temu, da cepivo ni toksično, ampak v resnici ljudje, ki so dobili to cepivo, niso imeli relapsov v času preizkušanja. In zaradi tega je zdaj v drugi fazi poskus vodi seveda ta newyorška najbolj znana onkološka bolnica, tudi ameriška Sloan Kettering. Cepivo je od BioNTecha in sej tukaj vidite že po članku, ne, da so to pravzaprav da so to njegovi raziskovalci. No in v tem trenutku se dela rekrutacija v Združenih državah, vendar se bo čez kakšen mesec delala rekrutacija tudi v 80 centrih po celem svetu, v glavnem v Evropi. Se pravi, to je nekaj, kar je drugi razlog, da je bila ta Nobelova res dobro izbrana. Seveda pa moramo bi spet rad opozoril, da videli smo vse tiste stopnje, ki jih je treba narediti. Pri takih stvarih, kot je Nobelova, se je najbrž treba tudi sprijazniti, da v resnici je deloma tista intelektualna inovativnost, tisto, kar se naredi znanstveno, pa tudi seveda okoliščine, ki pripomorejo temu. In pravzaprav se najbrž, razen pri prizadetih, ki jih človek razume, Roberta Malona jaz čisto razumem, da bi se mu zdelo pravično, da pravzaprav, če bi šlo za samo odkritje, da je tudi on tisti, ki je zraven, ampak seveda v življenju je veliko tudi igra na ključi in pravzaprav zelo človeško je, da z enakim veseljem, kot sprejemamo intelektualna veselja, je fino, da sprejemamo z enakim veseljem tudi te igre na ključi. Tudi to je del življenja, se pravi, pravičnost ni nadrejena življenju, ampak je samo del življenja. Vedno pa moramo imeti pred očmi seveda to ogromno verigo seveda vedno moramo imeti pred očmi to ogromno verigo raziskovanja, ki je pravzaprav bila pogoj, da nastane to, kar imenujemo cepivo proti COVID-19 in seveda, da je ta tehnologija, ki bo verjetno omogočala še veliko, veliko drugih dobrobiti v medicini. Hvala.
enako cepivo. Ne? Zakaj so pa potem spremenili to usmeritev, kako mi reči?
Aha, ja. Torej, bom rekel, nekako klasika je, zdaj, klasika je seveda, da je cepljenje, torej, eno je narediti cepivo, drugo ga je registrirati. In registracije so tiste, ki so v medicini odločilne. Se pravi, vse, kar je v okviru registracijskih študij in zdravniki delamo, smo nekako, se smatra, da samo uporabljamo zdravilo. Karkoli delamo, kar je izven registracijskih študij, pravzaprav delamo eksperimente, kar je po medicini zelo nezaželjeno, oziroma nihče ne želi, da se nad njim dela eksperimente, oziroma karkoli je narobe, pade odgovornost na zdravnika. Ampak seveda, COVID je bil poseben, se pravi, tu se tepejo principi, ki smo jih navajeni. Navajen smo, da če se cepi z enim cepivom, se dokonča to cepljenje, tako kot zahteva registracija. Zdaj seveda, vmes so se pojavili tisti zapleti z vektorskimi cepivi, ki smo jih nekako v Evropi, nekako smo nanje precej živčno odreagirali, ne? Druget niso, ne? Se pravi, Združene države so tako in tako uporabljale izključno Moderno, se sploh niso ukvarjale z vektorskimi cepivi, tam je bila Moderna oziroma mRNK zelo favorizirana. V Evropi smo bili do pojavov, se pravi, do neželenih pojavov, ki so bili sicer redki, ampak so bili pri vektorskih cepivih hudi, ne? Se pravi, te tromboze. Za te pojave se je vedelo, da se je hitro ugotovilo, da nastanejo pravzaprav pri pravzaprav ljudeh, ki imajo dve genetski posebnosti, ki sta združeni skupaj. Se pravi, ena genetska posebnost je, da imajo receptorje za adenoviruse, da so nenavadno reaktivni za adenoviruse, to je okrog 1–2% ljudi. Po drugi strani pa so bili to ljudje, ki so imeli še protitelesa, proti receptorju za heparin. Ta protitelesa pa tudi ima samo kakšen odstotek ljudi, in sicer so to protitelesa proti mikrobioti, zlasti proti ustni mikrobioti. Se pravi, ljudje, ki imajo recimo parodontalno bolezen, imajo pogosto protitelesa proti ustni mikrobioti, in ta protitelesa so hkrati tudi navzkrižno reaktivna z heparinskim receptorjem, zato ker so enaki polisaharidi. In ljudje, ki so imeli eno in drugo, teh je bilo malo, eden na 30, 50 ali pa 70 tisoč, ti ljudje so potem naredili tromboze. Ampak preden se je to zvedelo, je Evropa odklonila ta cepiva, in zaradi tega so potem začeli reči, ok, tudi če ste se cepili z vektorskimi cepivi, je sicer mimo vseh smernic, da bi premenili cepivo, ampak ker pa se zdaj bojimo vektorskega cepiva, je pa vsem boljše, da se ponovno cepite v tem smislu je bilo to. Se pravi, pač improvizacija, kot je med pandemijo.
Ja. Zelo lepo ste poudarili to povzročanje vnetja zaradi mRNA, ki je nemodificirana. Srečno naključje pri vsem tem je pa, da hkrati je pa taka modificirana mRNA tudi bolj stabilna. Če bi dosegli na drug način, da mRNA ne bi bila ne bi povzročala vnetij, koliko pomembno je pa to, da je ta modificirana mRNA stabilnejša, zaradi RNA v telesu bi verjetno zelo
Ja, zelo pomemben. Torej, v tem članku, ki ga nisem nadrobno razlagal, je pravzaprav tudi efekt, da je prav ta modifikacija povzročila tudi bistveno boljšo ekspresijo. Se pravi, v primeri z nemodificiranimi ne samo, da ne povzroči vnetja, ampak je tudi v članku en graf, da je ravno ta modifikacija pač zaradi tega boljša. In zaradi tega je ta patent, saj, ko govorimo o nagradah, smo potem blizu patentom. Se pravi, sami ste videli, da je takoj naslednje leto bil patent narejen, ki je bil potem odkupljen, da je bila ustanovljeno podjetje Moderna. Ne? In ko govorimo o patentih, je spet en del tiste igre usode. Ni vsako odkritje patentabilno. Se pravi, patent, ki ga je naredil Malone, ki je tudi naredil polno patentov, njegovi patenti so samo pokazali princip, ampak te mehurčke je mogoče delati na mnogo drugih načinov in veste, da patenti ščitijo samo točno določeno molekulo na točno določen način. Če ti lahko uporabiš neko drugo lipidno molekulo, potem patent nič ne velja, noben ne bo plačal za nekaj, kar lahko na drug način naredi. Če pa se najde modifikacija, ki pa jo ni mogoče drugo narediti, se pravi, namesto pseudouridina so marsikaj drugega poskušali, ampak ta pseudouridin se je pokazal kot daleč najmanj vneten in po drugi strani naredi daleč najboljšo produkcijo mRNK. Zdaj, tisti, ki bo šel ponovno odkrivati nekaj, bo mogoče lahko odkrival, ampak to sta že v tem članku naredila. Sej ta pseudouridin ni bil edini modifikator. Zraven je v tem članku še 5 drugih načinov. Sam od teh 5 drugih je pač ta pseudouridin daleč najboljši. Se pravi, tisti, ki bi hotel narediti boljše cepivo, bi mogel namesto tega pseudouridina nekaj drugega. In ker ni, je ta patent zaradi tega še vreden. Če bi bili drugi načini, ta patent ne bi imel seveda take vrednosti. In to so spet te igre usode, se pravi, tako da iz tega bo lahko veliko filmov še. Ja. Ja, zdaj, se pravi, to cepivo je bilo delano proti omikronu, ki ga seveda podrazličice, ki jo ni več, je bila poleti, ampak je ta podrazličica še vedno zelo podobna tem. Se pravi, cepljenje z sedanjim cepivom pravzaprav zelo dobro ščiti proti trenutnim omikronom, čeprav niso. Ja, nekdo, ki je prebolel nazaj pred manj kot pol leta, nima smisla cepiti. Tudi prevečkrat cepljenje je spet, opazili so en učink pri ponavljajočih se cepljenjih, zlasti če so preveč skupaj, da se pravzaprav dela neke vrste to, kar imamo pri alergologiji, ko delamo desenzibilizacijo. Torej, če desenzibiliziramo v alergologiji na ta način, da se velikokrat cepi antigen v kožo in če ima nekdo, naprimer, če je proti čebeli ali pa osi, ima IgE proti telesa, se s tem cepljenjem doseže, da najprej nastanejo IgG1 proti telesa, potem za ponavljajočim cepljenjem pa druga, pa IgG4 protitelesa. In ta IgG4 protitelesa so taka, ki ne aktivirajo komplementa in ne delajo opsonizacije, kar pomeni, da so IgG protitelesa, ampak nevnetna. In pri uspehu desenzibilizacije navadno gledamo kar indeks IgG4, IgG1. Ko IgG4 prevladajo nad IgG1, vemo, da je desenzibilizacija dosežena ne samo proti nastanku IgE-ja, ampak tudi proti vnetju, ki nastane zaradi IgG1. In tisto, kar se je opazilo pri večkratnem cepljenju, zlasti z Moderno, je, s cepivom Moderne, ki ima večjo odmerek mRNK-ja, kot pa od BioNTecha, ne, 3-krat več, se je opazilo, da je po večkratnem cepljenju ta prehod v IgG4. V resnici nihče klinično ne ve, kaj to pomeni. Lahko, kaj to pomeni za zaščito. Lahko nič kaj ne moti zaščite, teh študij še ni narejenih, se pa seveda marsikdo vpraša, ko pomisli na alergologijo, in je jasen, da se čim večkrat zaporedno cepimo in čim manjše so razdalje med dvema cepljenjima in čim večji so odmerki v posameznih cepivih, bolj zanesljivo bomo naredili preklop iz IgG1, ki ga več ali manj smatramo za zaščitenega, v IgG4. Zdaj, ta preklop se zakaj naredi? Zato, ker je telo narejeno na ta način, da hoče preživeti, in če je nek tujek stalno prisoten ali pa ponavljajoče prisoten, naredimo protitelesa, ki ne delajo več vnetja, ampak samo še vežejo na tujk. Zato, ker bi si s tem vnetjem več škode naredili, kot pa koristi. In zdaj, kot rečeno, to nenehno ponavljajoče se cepljenje, dejstvo je, da naredi več IgG4, od tu naprej bo treba pa še videti, a to kaj pomeni, lahko pa da nič ne pomeni za zaščito. Za čebelji pik je to super. Za čebelji pik je super že IgG1, zaradi tega, ker pri čebeljem piku dobimo anafilaksijo zaradi IgE, ki pa vedno nastanejo lokalno, ker so mastociti lokalno, in pri čebeljem piku je že super, da naredimo v krvi IgG1, ker potem, ko pride pik, sicer nastane velika oteklina na mestu pika, ampak ko pride strup v kri, ga tam polovijo IgG1 in ne pride več po celem telesu in ne more nastati anafilaktični šok. Še vedno nastane tip 1 lokalno, ampak to je buška. Ne nastane pa anafilaktični šok. Super pa je seveda potem, ko se IgG1 spremeni v... Seveda, IgG1 povzroča serumsko bolezen sicer. Če bi bilo velike količine, če nekoga popika 50 čebel, nastanejo imunski kompleksi. Kaj? Ja. Če pa bi imel že prehod v IgG4, potem pa niti imunski kompleksi več ne nastanejo, ki bi aktivirali komplement in delali vnetje, ne? Ja, pravzaprav je bila ta tehnologija kupljena zaradi HIV-a. Se pravi, Moderna je bila ustanovljena zaradi delanja cepiva proti HIV-u. To je bil osnovni namen, ker v tistem času je bil HIV zelo aktualen, ne? In glede na letence v medicini, ne vem, ni vedno, ampak v medicini je eno pravilo, ko se nekaj odkrije, če v 10 letih deluje, je je v redu. Če 10 let ni nič, ponavadi iz tistega ni kaj dosti. Ne velja vedno za vse, ne? Recimo, ne velja, naprimer, ker smo pravkar videli, da deluje presaditev, da so zdaj prvi pacienti, ki imajo presajena srca od pujsov. Ta projekt čiščenja antigenov iz pujsov, se pravi delanje kloniranih pujsov, klonirani morajo biti zato, da viruse ne prenašajo od svojih staršev, ne, ki bi bili smrtni za imunosuprimiranega človeka. Se pravi, delanje kloniranih pulsov je že 20 let. In v vsem tem času skušajo tiste najbolj antigene od pujsov, proti katerim se naredi največ zavrnitvenih reakcij, jih skušajo izničiti, pa seveda še vedno ohraniti živega pujsa. In ti projekti v nekaj skupinah, 5, 6 skupinah, to so ogromni projekti, ne toliko, kot je bilo to cepivo, ampak še vedno to so 100-milijonski projekti, ki potekajo že več kot 20 let. In v tem času smo se zaradi tega pravila pravzaprav že poslovili praktično od možnosti, da bo možno delati pujse kot vir za organe. Zato sem bil silno presenečen, da pa so zdaj vendarle prvi pacienti, ki so jim ta srca presadili, kar pomeni, da tega pravila 10 let ni vedno, ne? Ampak v primeru HIV-a bi pa rekel, osebno bi dvomil, da bo to veliko pomagalo, ne? Na srečo pa je HIV pravzaprav, nam rekel danes že banalna bolezen, ampak vsekakor je pa je možno zelo dobro jo kontrolirati.
Najlepša hvala. Če si imel še kakšno vprašanje, morda potem na odmoram, če bo profesor še z nami, se moramo premakniti naprej po programu. Tako da jaz bi še enkrat...
Ključek mora tle. Marko? No, right. So I will just continue by introducing Professor Vladimir Parpura. Okay. So I would like to introduce Professor Vladimir Parpura. We start with a new session today, and I will continue in English. Hopefully this will not be too much of a challenge, but it's basically all related to translation. Now we move over to the brain, to neuroglia, part of the brain that is also known as the other brain ali drugi možgani. Torej, Professor Vladimir Papura originally comes from Split. He did his career in the United States, and later he became a director of a huge translational center in China. He may introduce a little bit of that, and today he will talk about nanoparticles that are part of this development of translation efforts. As we have heard in the previous lecture, nanoparticles may be strong inducers of the immune response, but also a delivery of drugs. Vladimir, the podium is yours.
Thank you. Let me see. Good morning. Dobro jutro. I tell you, that will be all Slovenia you're going to hear from me. Last time when I was here, I gave, it was acceptance speech for Academy, and Nina told me that I speak light speed Slovenian, which was quasi-Slovenian, of course, anyway. So this is what I would like to do today. I would like to introduce you to these materials that we call carbon nanotubes. Rest assure you, let me see, where's the, oh, lovely, okay, good. This is a problem when you have, cheap multifocals, that's how we operate. Okay, so anyway, I hate multifocals, but here's rest assured, I'm going to present you this kind of driven by hypothesis. But as you may imagine, when new materials come in, everything that we think about is what I call squirt and try. You just put it on the tissue and you look at what is the effect. And that's exactly what you're going to see, how we came to some translational use in astrogliology, or if you want to say how the carbon nanotubes affect astrocytes. I'm not going to tell you much about Institute. I'm going to invite you to visit, if you wish, in Hangzhou, China. It's a small little town of 11 million people. We just annexed a little small town of 1 million people to it. I'm really proud of finally being recognized formally for collaboration with Professor Zores that's been going for, what, Robert, 25 years or something like that? Okay, and so we signed a memorandum of understanding because Chinese love these ceremonies of signing paperwork. Okay, anyway, so without further ado, let me first show you an outline of a talk. What I'd like to do is first tell you a little bit about what are the carbon nanotubes. In fact, what are the two modalities of carbon nanotubes? Carbon nanotubes is different, depends how you use it. And then translational applications that we know of. I'll give you a little hint on effects on neurons, because brain is composed not only in astrocytes. It's a wishful thinking, of course, on some of us, but neurons are there. And I'm going to give you a review of that and then show more detailed interactions with astrocytes and also astrocytoma, which is malignant astrocyte, essentially disease called glioblastoma multiforme. That is the most vicious cancer that we have in the brain. That later you're going to hear more from Professor Montana on different approach to translation approach. And then there will be some little envoy at the very end. This is how we can divide arbitrarily carbon nanotubes in their modalities, and that they can be colloidal solutes or they can be films. Now, the one thing that is important to realize that carbon nanotubes are not biodegradable. That can be a problem or can be a friend. I would like to think in this case not as a foe, but as a friend, that if you apply them, that you have prolonged effects and will not be degraded. Now, in terms of carbon nanotubes are essentially, these are single-wall carbon nanotubes. They're rolled up like cigarette paper that one would roll, right? It's a graphene sheet that is rolled up. It can be rolled up in different ways. We're not going to go into those details. They're wonderful soluble in compounds like everything that your brain would be dissolved in, like hexane and stuff like that. So in order to make it aqueous soluble, so colloidal solids in water, we need to add different compounds. Here it's para-aminobenzonic sulfonic acid is one example, which is citrionic, or polyethylene glycol. That is neutral, or we can then add tetrahydrofurfuryl, which is there really to prevent so-called hexagonal packing due to van der Waals forces in between single-wall carbon nanotubes. These nanotubes, at least the ones we use, are in diameter between 1 and 15 nanometers, which means that some are just single wall nanotubes, some are hexagonally packed. And the lengths are 0.1 to 2.2 micrometers that we use. Yes, they can be reproduced as a long streak in a different way, even a meter long, if it's needed. In terms of adding these functionalities, we add about 20% by weight in order to make it water soluble. So just that you give you a bulk number. The other way that we can use carbon, and this is how colloidal solute looks like. Essentially, it's a black liquid, very unappealing to your view. The one way that can be also used is so-called films. One can disperse it onto, for example, glass cover slips, make hot glass cover slips and disperse them onto it, and they actually retain there for months. We can make them obviously in different thicknesses, and you will see later how that is important. Here, anywhere between 10 and 60 nanometers. It looks almost like neutral density filter for those of you who are into microscopy. Okay, so let's look at now what are those applications of carbon nanotubes in so-called neuroprosthetics. Something that we approached already is they've used colloidal solutes in injury models of spinal cord injury in vivo, and that is in rat. TBI in vitro, you will see what is that simulation like for astrocyte culture. And then also in astrocytoma or glioblastoma. The other application is really how to make these electrodes implanted to brain better. Those of you who are into implantation for deep stimulation, you know that mainly use tungsten steel electrodes, which actually have Faraday conductances, which means they get dissolved by brain. So the electrode hurts, but the brain also hurts because it's not really nice to have heavy metal acting upon cells. So if we can coat these electrodes, that would be advantageous to less formation of scar, to less immune response, etc. What I'm going to do now, I'm going to show you a mix of a published data that dates back to perhaps 15 years up to unpublished stories. Our previous study shows with colloidal solutes, when we grow hippocampal neurons in culture on those glass cover slips, very artificial environment. You don't have any glass in your brain, but that's the best we can do. We plate neurons on a glass substrate, and we can visualize it that way. What we find out when we treat them with soluble carbon nanotubes, that their selected neurites extended much longer. So the reason why that happens without showing you data is there was a selective inhibition of stimulated endocytosis without affecting exocytosis. So a net influx of lipids into the cell plasma membrane and hence processes. So rationale then first is let's try to do it somewhere that it may actually accomplish something in medicine. One thing that one can do is, this is not quite normal unless you get cut by samurai swords, but here's a model that is used in adult female rats that we can do complete thoracic 9 transection. We form an injury which forms a cavity. Cavity is a problem in medicine because apparently nothing grows through it. However, when we apply here with gel carbon nanotubes, we saw an increase neurofilament positive tract fibers, which means the neuronal neurites outgrow through this what's supposed to be not passable through cyst. There was a decreased lesion in volume using Cavallari method, and also using base of Beatty-Brescian locomotor scale, there was enhanced locomotion. I don't have a movie here, but just to bring you in perspective, in this case, the rats would just move by using front limbs. In this case, they was also use rear limbs. The one thing that always is of nuisance is that injury always have an increased GFAP expression, which we call reactive astrogliosis. There's a good one and bad one. Now we are talking about one that can form scar. And it occurs around the injury. Carbon nanotubes have not affected that But in order to study that in more detail, we actually went into culture and looked at astrocytes grown on those same artificial substrates of polyethylenimine coated glass cover slips. And we can take here cortical astrocytes. These are from visual cortex, actually from mice of 0 to 2-day-old animals. And we treated for 3 days with water-soluble carbon nanotubes, be that PUBs or PEG solubilized. This data is with PEG, but similar is obtained with PUBs. We load then cells with calcine, which is very simple experiment. You load them with the dye that actually tells you that astrocytes are viable. It's in green, but doesn't matter. I don't see colors well, so we put it here black and white. Then you look at it, area, perimeter, and so-called form factor. What form factor is, if it's a circle, that's a 1. If it's close to a line, it's a 0. So what you can see here, there is increase of perimeter of cells treated with carbon nanotubes, and that results in a more form factor that they are elongated, kind of a little bit more mature, if you wish. Well, if you're going to say there's something immature, you might as well then look at what is the status of GFAP. Namely, GFAP is also an index of maturity of astrocytes, as opposed to being reactive astrocytes. So we looked at that by looking on effect on the parameters here. I define them here, what is density, content, occupancy. Bottom line is there is more GFAP by amount and coverage of the cell, as you can see when they're treated with carbon nanotubes. So one conclusion from that is that these cells might be more mature and they may be bigger than they were when we treated with carbon nanotubes. Well, if that's the case, in literature, there is observation that this might be associated with a change in expression on the plasma membrane of plasma lemma glutamate transporters. There are at least 2 kinds of that we entertain. There are many more. This is generalization, 2 kinds. There is glastin GLT. And in our case, these are cells that are from 0 to 2-day-old, and in our case, they get arrested developmentally when they were isolated from an animal, which means, if you trust any other literature, they will have a little to none expression of GLT, and most expression will be with glastin. Okay, to assess this, rather than doing these labelings, we actually use the primordially old method of radioactive detection, where you can take radioactive glutamate, you can imbibe these cells, here shown in green, they take it up, you wash it away, and you look how much of that radioactivity do you have. And you can see that in control situation, we have certain amount, which we call 100%. If we treated with functional group polyethylene glycol, there's no change in that. But once we put carbon nanotubes, we had enhanced uptake of glutamate. If so, if this is through these transporters, then we should be able to block it with a blocker of that, which is here shown as TBOA, and there's a block of that. Conversely, we should be able to enhance that. If we use, for example, ROCK blocker shown by Jacopo Meldolesi to enhance expression of plasma membrane receptors in the plasma membrane, we should enhance it. And here we go. If you use such a blocker, Y27632, don't ask me why it's named this way, there is an increase of uptake of glutamate. Bless you. Now, we did actually label, just for any case. We labeled here only plasma membrane component, not the entire component, by not permeable in cells. And you can see there is an increase of expression of that protein, in this case GLAST, when we have addition of carbon nanotubes that are soluble. So what might this have to do with tripartite synapse? Well, I know some of you expect that I'm going to come to tripartite synapse. After all, we introduced the concept of it, so I have to say something about it. But if you think about this astrocyte being close to a synapse, okay, here's a presynaptic neuron. This neuron, just for sake of argument, releases glutamate. Glutamate gets here a postsynaptic effect. Some of that gets spilled over, And astrocytes are here, sponges that are taking away that transmitter. They can also release transmitter. I mean, I'm not going to go into that. But if it's these astrocytes untreated with carbon nanotubes, that's how the situation will look like. If you would treat it, it will be increased perimeter and form factor would be decreased, which means it will be more stellate. GFAP would go up, glass would go up. So that's the difference. What do we expect to happen? So if this is next to the synapse, we'll mop it up more glutamate from the synaptic cleft in a situation that when there is an excitotoxicity. All right, so, okay, if that is the case, let's now entertain another idea. Let's entertain an idea that then carbon nanotubes that can actually increase production, that can increase expression of glass in a plasma membrane can be used for translational purposes in diseases or conditions that we know there is an increased expression of glass. And that is in traumatic brain injury. In TBI, the patients have decreased expression of GLAST, which means that glutamate is accumulating and aiding that injury. So if we can treat with carbon nanotubes, we might be able to jack that up and prevent the injury. So rather than going into brain, here's the model used. This is actually done mainly in Rijeka. Kristina Pilipović lab did this. What one can do is one can apply pressure onto cells that are grown on celastic membrane. Unlike glass, well, glass is a little bit deformable too, but this elastic membrane actually goes into large deformation, which causes a model of injury that people equate to that what occurs in vivo in TBI. Okay, if you look at that, if there is injury induced, there is no, there is some change in GFAP, but it's not statistically significant, so I call it no effect. And if carbon nanotubes are put there, there's no change there. What I was referring to is this expression of EAT1, which is glass essentially. We have here in TBI, there is reduction. That's what you expect from human data. And carbon nanotubes aid expression of to the level that is undifferentiated from that at the rest, which means if it's translatable TBI, perhaps then we can use this. How to apply throughout the brain that's a different story, but it is soluble compound, so you can apply on a site of injury. And this is the last of the attempt to use soluble carbon nanotubes, and this was one of those quart and try experiments when we use human glioblastoma cells, D54mg, that also express GFAP, so we don't need to put calcium anymore. We can ask ourselves, is there any change if we add carbon nanotubes or if we add their functionalized group? And what you can see is that this is different than we've seen previously in neurons and astrocytes. Previously, the effect was driven by carbon nanotubes backbone. But this data tells you that basically the whole effect that we see here from soluble carbon nanotubes is due to the presence of the moiety, THFF moiety. So this is unintentional effect of the added compound to make it soluble. That's something to keep in mind, because people get really excited about translation and they don't do proper controls. And then you can find here that actually the functionalization group is the one that has an effect, not the backbone. So let's look at it a little bit further. One thing of interest in those of you who are into clinical enterprise is that, yes, GFAP is expressed, especially mRNA in glioblastoma, but it's dwarfed by expression in astrocytes. It is the best marker in GFAP that you use in clinic, but when you compare to astrocytes, it's really almost non-existent. Okay, so the one issue with glioblastoma is the proliferation ability and adhesion ability, right? So what we can do is we can look at it naively by putting cells on a substrate. You can see them here in green. You can actually label their live nuclei, and you can look at whether the cells have both. Those are alive. If the cells has only red, the cell is dead. Not red, but blue, I'm sorry. If it says only blue, it's dead. So here's, it's a little bit busy slide, but let me lead you through it. You're looking at the 2-hour number of cells as you plate them, which we call adhesion. You can see it's similar regardless whether you treat them with carbon nanotubes or not. And it's actually similar regardless whether you treat them with functionalized groups. Now, what you can do, you can wait now for 2 days, and you can see that now there is an effect of a carbon nanotubes with PEG. And this is actually similar to effect, and that is driven by the backbone, but not with the functional group. If you look at percentage of dead cells, there is an increase in dead cells when we treat with carbon nanotubes PEG, THFF, but that effect is actually opposite when you use only the group. So which means that functional groups may have actually protective effect. In this case, if you add this functionalization group to deliver it to the tumor, you are actually reducing the effect that carbon nanotube backbone would have. In fact, it does here increase the cell death. So this is the first modality. The second modality is that of brain-machine interfacing. And here's the issue. I already mentioned you can put here an electrode, deep stimulation. We use it all the time for Parkinson's disease, for instance. But there is a formation of scar there. And as I said, the major issue there is the issue of being chewed up by the brain, because the brain actually is a salty solution. So steel gets chewed up, and that causes the injury of the brain and deteriorates the electrode. Kiefer attempted to avoid this issue by coating these electrodes by carbon nanotubes, and actually those did help and had better performance and longer performance in at least in a rodent brain. The issue becomes is, what is the quality control of those electrodes? Namely, how much of carbon nanotubes should we put in? What should be roughness of those carbon nanotubes, and what should be conductivity? Just remind you, carbon nanotubes can be almost insulators to superconductors. So somewhere in between, we have to find ground how to coat it to be valuable. We initially attempted this by using neurons, and if you look at I gotta take now glass cover slip PI as a standard, although I gave you that statement there is no glass in your brain, but that's our initial conditions. If we now vary the conductance of the substrate without changing the roughness, we can see that if we increase conductance to 42 Siemens over centimeter, that is similar looking like in glass, while in between conductances, they are lower, may change the size of a body, for instance, and the neuron length. And there's change in number of growth cones. So knowing that, we can now ask, what is the situation with these conductances with astrocytes? Because after all, those are the cells that will be forming the scar. And in this case, we don't worry too much about roughnesses. We let roughness go together with conductance, and so we are looking at as a composite change in material. You can see them here under atomic force microscope. There's an increase in thickness, there's an increase in roughness, and here measurements of conductivity. When we grow astrocytes on those strata, you can immediately see, I mean, you gotta be kind of a little bit blind not to see the effect, right? This is normal glowing polyethylenimine. They kind of look more ramified. This looks like a typical fried egg, if you wish. And you can see that by an increase in area and becoming more round based on the form factor. So for those of us who are sometime in a field, we immediately worried about this becoming dedifferentiated cells, which means that GFAP might be downregulated, and that is the case. So these are bona fide dedifferentiated astrocytes, which which is called B cell by Arturo Alvarez Buya some time ago. So those cells can proliferate and provide new cells. So that's what we tested here. Again, adhesion, proliferation, and this is a graph. You can see that keeping here for 4 hours, there is a certain adhesion. If you keep them for 4 days, they proliferate. Of course, not like a glioblastoma, you know, not even comparison. Little proliferation is there. There's no change in the depth. But what we've seen is that when we grow them on more conductive and rougher surfaces, there is a disproportionate increase in proliferation of these cells. So now you can make some judgments call and recommendations on what might be the appropriate coating of those electrodes. We would suggest, by comparison to standard for us polyethylene, that 60 nanometer film, which is 42 Siemens over centimeters, is the best because it retains the morphological properties of neurons, but is actually offering neural stem-like cell property of astrocytes. And that's where we're gonna stop. These are conclusions that we can use both modalities in translational. I remind you, this is still in a crib, right? This is the new materials that it's a squirt to try. So we need to find out what are those safety measures. In fact, no government yet declared what is the safety measure of using these materials, although they're in use quite a bit. For example, in golf shafts, for those of you who are golfing. This is the lab in the United States. Vedrana is the only person still with me. She'll be giving talk later on. The collaborators, and this is late Robert Haddon and Elena Bekiarova, Candice Floyd, Mike Brenner, and lately Kristina Filipovic in Rijeka. And this is how the lab looks like today in Hangzhou, China. This is on the campus, you know. We just have affection to Hangzhou and campus. So this is iLab's ECMU thing. The reason going to China, obviously, there has to be a reason, right? Money is a really good reason to go somewhere. And the amount of funding that I received from Chung Kong and Kung Peng Action is just staggering. So I would like to thank them too. If you use WeChat, you can connect me this way. If you use email, this is this way. There's plenty of rooms for any type of joining if you wish. If you wish to join a graduate student, the audience is a little bit too mature for that. Or if you want to join as a postdoc, some of you look that way. Or as a faculty, we can handle that. At this point of time, I just like to thank you for your attention, and hopefully I did not put you to sleep. Thank you.
Thank you, Vladimir. Questions? So with these nanotubes make astrocytes angry?
So you know, why it might be, you know, what I didn't go into here, we actually looked at that. It looks like it's a structure of nanotube. The way we make them is just proper width and length to wedge the vesicle. You know everything. Robert knows everything we need to know about fusion pore. But there's a certain width of fusion pore, and these nanotubes can actually go inside there, and because of the length that we provide, the vesicles that are 300 nanometers in astrocytes cannot pinch off anymore. So there's a block of endocytosis. And it looks like that. Why did they bind there? I have no clue. Okay, I'm working with the hypothesis there's just a fluid phase endocytosis, but it doesn't hold properly because if you look at endogenous endocytosis versus stimulated, they prefer to block stimulated endocytosis. So why there's this attraction to it, you can speculate there might be the van der Waals wall that around that gives a negative cloud, and maybe this is interacting with phospholipids. But you know, that's a long stretch. You know, that's bullshit galore right there. But anyway, it's for mental exercise. It's something that you can convince yourself that might be the case.
Thank you, Vlad.
Thank you. Oh, Yanay. Yanay, you have to ask. Yanay, how you doing?
Great, thanks. Thanks for the great talk, Vlad.
Thank you.
I have a question regarding your application with electrodes. You also mentioned
I heard vacation?
the
Oh, application. So I heard vacation and said, yeah, finally I'm taking one.
no, not yet. You also mentioned they are not biodegradable.
Correct.
So should we be worried they would be causing some sort of long-term effect?
Absolutely, you should be worried. That's what I'm worried about, and I try to convince U.S. government to go into investigation of those toxicity levels and to give some money support for that. But they said, that's a great idea, why don't you do it? But we are not going to support it. So we do what we do, but it should be, those studies on toxicity require long-term, as you said, require a lot of animals and require a lot of people to get involved. I don't understand European Commission, I don't understand United States government, and I have not understood yet enough Chinese government to even talk to them about this. But it is something that absolutely should be tested. The industry is booming. As I said, you have them in golf shafts. I mean, how would you like to hold that shaft knowing there's a material that might actually kill something in your brain and you have no clue what it's doing? But that's what we're doing. It makes great golf shafts that you can hit the ball much further with better control. But absolutely we should be concerned. We should be concerned about lungs if we breathe it in. Is this going to be another asbestos fiasco, which I'm very sensitive to because, as you know, there was asbestos disaster in Vranjic, which is next to Split, that we end up being in the movie. If it ends up being like that, that would be terrible. It is somewhere, once they're in liquid form, it's less worry than that is aerosol. Okay, and the exposure data in papers varies from, this is the worst shit you have ever seen on the planet Earth, to this is so benign that you can drink it easily and inhale it easily. So that's what is the spread, everything from 0 to 100. Where's the truth? I have no idea.
Thank you, Vlad.
Thank you.
So now we will continue to these presentations by our junior colleagues, and the talk that I would like to present now is by Urška Černe. Urška, where are you? So she will open up a new view of how we can actually learn about our brain through Vinska mušica, Drosophila. It's a preparation that is omnipresent with the climate change. We will also have a challenge to actually have Vinska mušica Drosophila all year round when you put fruit on the table. But this is much more we can learn. And Urška is finishing her PhD, and she will be great to actually introduce the Drosophila models into secondary schools, I guess.
Yes, thank you, Professor Robert Zart, for the introduction. So I will present my work that we actually did together during my PhD. So first, I will just like to make a short introduction about noradrenergic signaling in our brains, especially in astrocytes, and let's say present some motivation why we are actually doing experiments on Drosophila. So astrocytes, as you probably know, are a subtype of glial cells that provide metabolic, homeostatic, and structural support to the neurons. And astrocytes are believed to be the main target of neurotransmitter noradrenaline. So when noradrenaline binds to adrenergic G protein coupled receptors, it causes changes in intracellular calcium and cyclic AMP levels. So, for example, when noradrenaline binds to alpha-1 adrenergic receptors, it activates phospholipase C and increase intracellular level of calcium. And for example, when noradrenaline binds to beta adrenergic receptors, it activates adenylate cyclase and increase cyclic AMP levels in the cells. So in astrocytes, noradrenergic stimulation is also considered to increase extracellular glucose uptake, glycogen degradation, and lactate production in a process of aerobic glycolysis. This lactate can be then transferred through transporters to neurons and used as a fuel in oxidative metabolism. So it's well established that noradrenaline has important role in many cognitive processes like attention, learning, memory formation. And malfunction of this noradrenergic system and reduced glucose utilization are actually hallmark of aging and many and neurodegenerative diseases. So the question is, does these changes in glucose metabolism actually occur due to the malfunction of noradrenergic system, and which, as I said before, controls the brain metabolism, and how this contributes to cognitive decline. So now let's go back to Drosophila. So you probably all know fruit fly, Drosophila, this annoying little creature. And we used Drosophila as a brain aging model. So in Drosophila, there's no noradrenaline, but octopamine acts similar. It's a homologue of noradrenaline, and it modulates metabolism, behavior, learning, and also memory formation. And it also binds to octopaminergic G protein coupled receptors and causes changes in intracellular level of calcium and cyclic AMP. So in our aged Drosophila model, we actually observed neurodegenerative vacuoles marked here with yellow, and we also observed reduced locomotion behavior, which was tested with climbing assay. So our aim was actually to determine if aging affects octopaminergic regulation of cell signaling and metabolism in Drosophila brain. So to test that, we design experimental flight that so we did that with crossing a driver line that carries cell-specific promoter and transcription factor GAL4 with another line, reporter line, which carries gene of interest, in our case genetically encoded nanosensors for calcium, cyclic AMP, lactate, and glucose, under control of upstream activating sequence, UAS. And then in F1 generation, this transcription factor GAL4 binds to UAS and promotes expression of gene of interest, so in our case sensor. So then we age this F1 generation of flies, and dissect the brain, the whole brain, and did the ex vivo whole brain imaging with confocal, real-time confocal microscopy upon octopamine and also glucose and lactate stimulation. So this is our results. Here on these first two figures, you can see expression of calcium sensor in astrocytes, cortex glia, which is another type of glial cells in Drosophila, and in neurons. And as you can see, octopamine increases calcium signals in all three cell types. But in aged Drosophila, in aged brains, we didn't detect calcium increase. So this suggests some age-related impairment in calcium signaling in Drosophila brain. Further, here you can see expression of sensor for calcium for cyclic AMP, and stimulation with octopamine also increases cyclic AMP levels, but only in neurons. And these changes in signals was actually the same in young and aged brains, so there was no differences. But we didn't detect any changes actually in glial cells. So to investigate this further, we actually stimulate the brain of Drosophila with forskolin, which is activator of adenylyl cyclase, and here you can see that forskolin actually increase cyclic AMP only the same as octopamine, only in neurons, but not in astrocytes. So there is apparently different cyclic AMP signaling in Drosophila upon octopamine stimulation. Then we want to see if astrocytes and neurons are able to uptake glucose and lactate. So we stimulated the brains with elevated glucose and lactate, and here you can see that both astrocytes and neurons can uptake glucose, probably through glucose transporters, and that this glucose uptake is actually much reduced in neurons. On another hand, stimulation with elevated lactate concentration did cause the increase in intracellular lactate, suggesting to increase to uptake of lactate through transporters, but there was no differences that we detect in young and in aged Drosophila brain. So then we also stimulated the brain with that express glucose sensor and lactate sensor on the next slide with octopamine, and octopamine actually increases intracellular glucose only in astrocytes but not in neurons, and this glucose increase was actually reduced in aged brains. So in the case of lactate, octopamine increased intracellular lactate concentration only in neurons, but not in astrocytes, and this indicates activation of aerobic glycolysis only in neurons, most likely via cyclic AMP signals. And we also didn't detect any changes between young and aged brains. So to conclude, as we saw, octopamine triggers intracellular calcium signals in all 3 cell types, so in astrocytes, cortex glia, and neurons, but activates cyclic AMP signals only in neurons. Neurons, but not glial cells, exhibit octopamine-induced aerobic glycolysis in Drosophila brains. And in Drosophila, there is an impairment in octopaminergic regulation of first calcium signaling and second glial glucose uptake, as well as reduced delivery of glucose to neurons. So together this may contribute to the age-related neurodegeneration and locomotion behavior that we saw on the first few slides. So I would like to take my supervisor Nina Vardian and Annemarie Horvat and Nika Kozoderc, who also did some experiments in this study. I would also like to thank all my co-workers in the lab, and also you for your attention. Thank you.
Thank you, Ruška. Questions? So how can we stop the changes you have measured as a function of aging by taking not drugs, but perhaps nutrition?
So how can we change changes?
Slow down the aging.
Yeah, that's the good question, which I don't know yet the answer. Yeah.
But is that known, like, that, I mean, dopamine doesn't pass gut barrier.
Dopamine, no.
Yeah.
But there are many nutraceuticals that actually are heavily advertised, and they may also, be a useful sort of, perhaps a playground to actually test on your model whether they actually alleviate the problems of climbing essay. That is a learning essay in Drosophila.
Yeah.
And so that is something very, very important. I can see many of us thinking about, what is going to happen in the next 5 years, next 10 years. How can you stop that? And of course, Vlad knows that.
I don't know anything. This is what I'm asking now. I can't remember now from medical practice, but do we know the relationship between, like patients with some issues with dopamine, such as Parkinson's?
Well, I don't know much about dopamine, but the next speaker actually will talk about it, so maybe he will answer your question.
Yeah, so now we're in trouble.
Sorry, Keita.
But it was related to lactate.
Maybe we should.
Yeah.
Thank you, Ruška.
Thanks.
And the next speaker is going to be Keita Sugiyama. And he will talk about dopamine. And here you have this, yes. Keita is now living in Ljubljana with his family, and he will present himself.
Oh, okay. I don't know, just I'm speaking English. Oh, sorry. Oh.
This way. And this will go forward.
Okay.
And this is in between. This is the pointer.
A pointer, okay. Yeah, thank you for your kind introduction. Dober jutro, everyone. Today, I'd like to talk about my research here. I am Keita Sugiyama, a postdoctoral researcher at the University of Ljubljana. Prior to coming to Slovenia, I was in Department of Pharmacology, Kurume University School of Medicine in Japan. Here's a picture of my family, my son, and my wife. We are originally from Kurume City in Fukuoka in Japan. and moved to Ljubljana in April. My research focuses on dopamine, a neurotransmitter. In this research, I aim to understand how dopamine works to develop new treatments. Most studies in this area have primarily focused on dopamine's effect on neurons, leaving out of the role dopamine plays in non-neural cells like astrocytes. This knowledge gap led me to focus on elucidating dopamine's impact on these astrocytes. To deepen my understanding, I moved to Slovenia and joined Professor Zorek lab. Now, let's move on to the discussions. To provide some background, dopamine is an essential neurotransmitter that plays a critical role in regulator motor function and the reward systems. Dopaminergic neurons project to various brain regions, including the prefrontal cortex and the striatum and the neck. Generally, dopamine affects brain functions by acting on two types of receptors, D1 receptors and D2 receptors. Astrocytes play a key role in homeostasis and defense within the central nervous systems. They have dopamine receptors in several brain regions, including the striatum, neck, and the cortex. When these receptors are activated, it affects intercellular cyclic AMP and free calcium levels. However, much remains to be understood regarding their detailed molecular pathways and functions. Recent study shows that astrocytes are key elements in dopamine D1 receptor signaling, indicating that they will play critical roles in synaptic regulation in the reward system. Therefore, astrocytic D1 receptors may be potential novel cellular targets for targeting neurological disorders. Astrocytes also play a key role in energy metabolism, particularly in converting to glucose to lactate through a process called aerobic glycolysis. This metabolic pathway is strongly influenced by noradrenaline through adrenergic receptors, which affect intercellular calcium and cyclic AMP signals. Although the disruption of astrocyte metabolism has been linked to various neurological disorders, there has been linked to various there has been limited research focusing on the role dopamine in astrocyte metabolism. The primary motivation of this study was to investigate how dopamine regulates astrocyte metabolism, focusing on its actions through D1 receptors. Dopamine is known to increase cyclic AMP production through D1 by acting on these D1 receptors. Moreover, some D1 receptors can induce elevations in calcium levels. In this study, we first focused on cyclic AMP signals, measuring the intercellular concentration of cyclic AMP to explore dopamine's effect on astrocyte. Then we looked at lactate levels to see if dopamine regulates aerobic glycolysis in astrocytes. Next, we used the flat-based cyclic AMP biosensor, EPAC1 CAMPS, to study cyclic AMP dynamics. This sensor measures the relative fluorescence of two fluorescence proteins, CFP and YFP. We then conducted ratiometric imaging of CFP and YFP fluorescence, presenting the result as the typical CFP-YFP ratio traces. And here, I show you the experiment designs. First, we prepared the primary astral cultures from the cortex of oyster rats. We then transfected cells with the flat cyclic AMP sensor After transaction, we treated the astrocyte with different concentrations of dopamine. Oh, okay, I'm sorry, I was able to see the expression D1 receptor but on my laptop. But as you can see, the figures show that the primary large astrocytes express D1 receptor. Here's the result. To investigate the effect of dopamine, we established a dose-response curve, finding a dose-dependent response in astrocytes to dopamine as seen CFP-YP ratio. The half maximum effective concentration was 0.5 micromolar. Simultaneously, we created responsiveness table of astrocyte to dopamine. When using a 10 micromolar and above, every astrocyte we monitored showed an increase in cyclic MP. Therefore, we decided to use 10 micromolar dopamine and 100 micromolar dopamine to further investigating the cyclic MP increases. With the addition of selective D1 agonist, SKF, when we obtained an exponential increases in the CFP-YP ratio signals. However, when compared to mean time constant, the increases in the CFP-YP ratio was faster with 100 micromolar dopamine than with SKF. This suggests the dopamine increase intercellular cyclic MP by activating D1 receptors and possibly other receptors. Here the effect of D1 receptor antagonist and the beta receptor antagonist. With the addition of SGH and dopamine, we observed the effect of 10 micromolar dopamine was inhibited, while the impact of 100 micromolar dopamine was not. Next, we pretreated cells with propranolol, a beta receptor antagonist. The effect of 100 micromolar dopamine was inhibited more significantly than that of 10 micromolar pretreated SGH. Additionally, when SGH and the propranolol were combined, the effect of 100 micromolar dopamine were completely inhibited. This indicated that 100 micromolar dopamine increases intercellular cyclic MP through both D1 receptors and beta receptors. Next, we used the FLET-based lactate nanosensor, laconic in isolated cortical astrocyte, to measure the intercellular lactate levels in real time following exposure to 100 micromolar dopamine. As you can see, there was increase in lactate in 100 micromolar dopamine. In contrast, the control experiment shows relatively high responsiveness to the vehicle, indicating the level of sensitive astrocyte to mechanical stimulation. These are our findings. Our results suggest that the dopamine modulates intercellular cyclic MP via not only the D1 receptor, but also through the beta receptors. And that dopamine facilitates aerobic glycolysis via these receptors. Currently, we know that the mechanosensory signaling in astrocytes evoke calcium responses. Further, we found that an increase in calcium is a primary trigger for aerobic glycolysis. Therefore, there is a need to monitor calcium levels. Here are our next steps. We plan to further investigate the dynamics of intercellular calcium and glucose in astrocyte to elucidate dopamine's regulatory role in astrocyte metabolism. In furthering our research to the pathophysiology of neurological disease, particularly major depressive disorders, we plan to use liposaccharide LPS to mimic a mouse model of depression that involves inflammation-associated depressions. And then examining the changes in dopamine's regulatory mechanism in astrocyte metabolism. To translate our findings into in vivo experiments, we have formed collaborations with universities in Japan. Ultimately, our goal is to elucidate the fundamental insights regarding the function of dopamine in astrocytes toward development agonists or antagonists that mitigate the signaling mediated by dopamine in major depressive disorders. I would like to thank my co-workers who have contributed to this work. That's all I have to say. Thank you for your attention.
Thank you, Keita. Vlad, just a quick one.
Yeah.
Yeah.
It would be really intriguing to go in to look at cocaine. Cocaine addiction and speed. Ketamine, because you might actually have, some issue with lactate changes as well.
Oh, okay. Thank you. Thank you for it.
And then another associated thing is we're interested to look at association between various forms of stroke. And patients with addiction versus patients with, let's say, Parkinson's.
Parkinson's.
Some data exists in it, I just can't remember.
Yeah, thank you.
To relate more to
Thank you for your suggestions. I will talk to you later. Thank you.
Thank you. I would thank you, Keita, because we need to actually catch up a little bit. So thank you very much.
Okay.
And we have, before the break, we have another lecture, and it is by Zala Smoulin. And she will talk about G proteins. What are G proteins? G proteins, most of the drugs we take are targeting G proteins. But there are about 100 genes that are called orphan, sirote. We don't know what is their function in the body, and today Zala will talk about one of these, and we hope is going to be a promising new target for neurologic indications. Zala.
Thank you very much, Professor, for your introduction. I would also like to thank Slovenian Physiological Society to give me the opportunity to present our work here today. So I will talk about the density of GPR27 receptor in astrocytes and leukosteroidal neurons, and also if this receptor is involved in modulation of intracellular lactate inside these two types of cells. So we've heard quite a lot today about the metabolism in astrocytes. Astrocytes and neurons are metabolically coupled, and in the active parts of brain where neurons are very active, we can observe a rise in blood circulation and also in glucose consumption, and unproportionally lower rise of oxygen consumption, which means that glucose must be metabolized through glycolytic pathways. We've also heard about this already today. The process is called aerobic glycolysis, and in the central nervous system it occurs mainly in astrocytes, right here, resulting in big production of lactate. And according to astrocyte to neuron lactate shuttle hypothesis, this lactate is then trafficked from astrocytes through MCT1 or 4 transporters, which have low affinity for lactate, into the neurons, seen here, through MCT2 transporter, which has high affinity for lactate. This lactate can be then turned back to pyruvate that can then enter mitochondria for oxidative phosphorylation and energy production. It has already been shown that noradrenaline, which is mainly produced in locus coeruleus neurons, but also extracellular lactate, are potent activators of aerobic glycolysis inside astrocytes. They both bind to GPCR receptors on astrocytes, causing a rising or activation of aerobic glycolysis and, of course, a big release of lactate that can be then, as we said before, trafficked to neurons for oxidative phosphorylation, but it can also backfire locus coeruleus neurons to release even more of noradrenaline. So this effect of lactate on locus coeruleus neurons, but also the fact that adrenoreceptors are also present on locus coeruleus neurons, that has been already shown before, raised the question if maybe those two agonists also have an effect on the metabolism of intracellular lactate inside locus coeruleus neurons. And if so, is this metabolism different than the one seen in astrocytes? Recently, another receptor, GPR27, which is an orphan GPCR receptor with unknown functions, was proposed to modulate ERK and AKT signaling, both augmenting aerobic glycolysis. So another question that, of course, arose was, is maybe this receptor present on astrocytes, or maybe even on locus coeruleus neurons? And if so, does it affect the intracellular metabolism of lactate? We know that astrocytes are able to store glycogen in contrast to neurons, and are able then to rapidly activate aerobic glycolysis and lactate production. So we suggested that in our experiments, the metabolism or metabolic handling of lactate is going to be different in the two cell types. Our first aim was to investigate the effect of lactate, extracellular lactate, and also noradrenaline on the dynamics of intracellular lactate changes in the two cell types. And in the second part, also to investigate the possibility of presence of GPR27 on the two cell types and how it is involved in metabolism of lactate inside the two cell types. To do that, we first prepared the primary cultures of locus coeruleus neurons and cortical astrocytes from 1 to 3 days old rat pups that we then immunochemically stained using primary antibodies to show GPR27 receptor, or we transfected them with a FRET-based sensor that we also seen before in Keita's presentation called Laconic. We used confocal microscopy for these experiments, and we obtained the signals from donor and acceptor protein of the sensor, and plotting them against each other gave us relative changes inside the cells, how the lactate changes in the two cell types. So upon addition of extracellular lactate in 10 millimolar concentration, we observed a rise in intracellular lactate in all of the cells tested, and you can nicely see here in the panel A how the relative changes inside neurons and astrocytes are happening. We observed that that lactate elicited higher responses compared to control experiments in both cell types, but was much higher in astrocytes compared to neurons. In the beginning of the response, the speed of lactate change or the rate of lactate change was not significantly different, but it was in the later phase, especially in this part of the response that we see here and here, and this aligns very well. So, moving to the noradrenaline results. Upon addition of noradrenaline in 100 micromolar concentration, we've seen an increase in intracellular lactate in 70% of neurons and in 86% of astrocytes. But in contrast to lactate, noradrenaline elicited a higher response in locus coeruleus neurons compared to astrocytes. Here we did not observe any significant differences between the two responses regarding the rate of increase of lactate inside the two cell types, which aligns very well with what we can see here in the panel A. This suggests that noradrenaline might have a self-regulatory loop on the locus coeruleus neurons. Okay, as I promised in the second part, we are investigating GPR27 receptor, and using immunocytochemical identification, staining, we identified the receptor on both locus coeruleus neurons and also on the astrocytes. You can see the signal in green here and here. We identified locus coeruleus neurons using primary antibodies against dopamine beta hydroxylase, which is an enzyme that is expressed in adrenergic neurons, and astrocytes were identified using primary antibodies against GFAP glial marker. In blue, you can see the nuclei stained with DAPI, and DIC presents transmitted light pictures. And in the panel B, in both pictures, you can see control experiment without primary antibodies against GPR27. We analyzed the density of GPR27 receptor on the two cell types, and we expressed it as GPR27 positive area in regard to whole cell area, and at 20% threshold from the maximum intensity of the signal, we can see that the density is much higher in neurons compared to astrocytes, and also that all the neurons expressed the GPR27 receptor, but only 90 out of 217 astrocytes were found to have this receptor in our study. So then we asked ourselves, how can this receptor modulate intracellular lactate if it does that? To test that, we used two agonists of the receptors of this receptor shown here in the panel. And the first one was called 8535, and the second one 8535N. And as you can see, the structure of both agonists is quite similar. The only difference is here in this group right here. On the left, we have carbonyl group, and here sulfonyl group. In these experiments, we can see, we shown that the responsiveness was for both agonists higher in locus coeruleus neurons compared to astrocytes, possibly explained by the different densities of the receptor. And in average, the maximum increase of intracellular lactate was much higher for both agonists compared to the control groups in both cell types. But the most potent response we've observed in astrocytes upon addition of 8535 agonist. In the beginning of the increase of intracellular lactate, we did not observe any statistically important changes, but we did observe them here in the later phase, and we see that indeed 8535 in the astrocytes was the most potent activator. The rate of increase was the highest in this experiment, which aligns very well with what we can see here in the panel A. So to conclude, extracellular lactate and noradrenaline are involved also in the intracellular lactate regulation in the locus coeruleus neurons, not only in astrocytes, and the dynamics in astrocytes and neurons differ significantly in the presence of these two agonists. We've seen that in contrast to extracellular lactate, noradrenergic stimulation elicits higher changes in locus coeruleus neurons than in astrocytes, and noradrenaline might have a self-regulatory role on locus coeruleus neurons. In the second part of our work, we've shown that density of GPR27 receptor appears to be much higher on the surface of neurons compared to astrocytes, and this receptor appears to be involved in the regulation of intracellular lactate metabolism in both cell types. Changes are, however, observed in astrocytes compared to neurons, and the most potent agonists seem to be 8535 of the two tested. To finish this presentation, I would like to thank my co-workers in LNMCP group, especially to my mentor Helena Chowdhury and co-mentor Marko Kreft, and also to the professor Robert Zarec, also to the guys who supported this financially or in other ways, and last but not least, thank you all for the attention.
Thank you very much. Thank you very much. Any questions? I would suggest we have the questions on the other side of the square. Prešernova dvorana, it's in the ground floor, and where is the mini coffee? So Zala will be happy to explain to you, how this relates the coffee to
And
the
happy to drink coffee.
and cycling and peace. Of course, so that is we are going to be back at 12. So we have a longer coffee break, so until 12. So we are invited to come here, so we then continue with the next session. So Zala, thank you. Thank you. Welcome back. So the topics that we will discuss in this second part will all relate to some form of pathology. And basically the brain that actually responds to pathology, probably in the first line, is not the neurons, but is astrocytes or glia. So the first part of pathology related will be to glioblastoma, which is a intracranial tumor, very common, and very likely to be treatable in the next future. At the moment, the prospects are very, very grim. And the first speaker will be Vedrana Montana. She comes originally from Rijeka, but she made a career in the States and now she works in China, and in particular in the domain of translation, how to actually bring the data that we develop to the actual use for the patients. And Vedrana, the podium is yours. And this is a pointer.
Dobar dan. Good afternoon. It's already afternoon. First of all, I would like to thank Robert for giving me this opportunity to share the research that we do in International Translational Neuroscience Research Institute in Hangzhou, China. So I will tell you a long journey from bench to bedside in about 20 minutes. This is the project that I started when I was working with Harry Sonheimer, then brought it in Vlad's lab, and now it's one of the projects at the Institute. So I will give you a little background, and then I will talk about the role of bradykinin and bradykinin receptor in GBM, glioblastoma multiforme, and how bradykinin 2 receptor emerges as a target for therapies. So in central nervous system, we recognize two types of tumors, primary tumors that originate from the cells that reside within the brain, and that's how they are named, or secondary or metastatic tumors, which usually are metastases from tumors that originate in the other parts of the body, like lung, breast, melanoma, colon, etc. Today I'm going to focus on glioblastoma, which is grade 4 glioma. That tells us it's one of the worst, the deadliest, and indeed, together with pancreatic cancer, it's one of the deadliest cancers that we know of, and it's untreatable. Glioblastoma has very unique environment. Unlike other tumors, it grows in very confined space, and usually forms this so-called satellite formations. So this would be a primary lesion site, and then there are additional lesion sites that form within the brain. As far as we know today, it does not metastasize. However, it does proliferate, migrate, and infiltrate in healthy brain tissue extensively. Standard care for those patients is surgery, if possible, followed by radiation and chemotherapy. And despite those aggressive therapies, median survival for those patients is only 14 months. So, as I mentioned, those tumor cells, they like to spread within the brain. There are several pathways where they, how they spread. They like to follow tracks, especially along blood vessels or along white matter tracks. What I will focus on more is their spread along blood vessels, and that pathologies has been repeated in experimental conditions, both in acute slice or in mice when they were implanted brain tumors. So what actually happens during that migration or invasion along blood vessels? Well, as we heard a lot this morning about astrocytes, one of their function is to attach or unwrap their so-called part of the cell that we call end foot around blood vessels, and they regulate exchange of nutrients and gases and permeability of blood vessels in the brain. Well, when these invading glioma cells start crawling along blood vessels, it actually disrupts that very tight blood-brain barrier, which leads to loss of tight junctions, degradation of basement membrane, and then leakage of serum into parenchyma. And I would like you to remember this disruption because it's important for the story a little bit later. So glioblastoma are known to be very heterogeneous tumors. They are heterogeneous between people, very different between people, but also within tumor itself. They have lots of different kind of cells. The cells that are growing on the edge of the tumor, they are of course in contact and interaction, in talking to normal brain cells like neurons, oligodendrocytes, microglia, and then of course astrocytes and capillaries. And I will focus this talk a little bit more of this part with this peripheral tumor cells that like to leave primary tumor and interacts more with the blood vessels and astrocytes as well. In about probably last 15 years, it has been discovered a cell that is named glioma stem cell, or brain tumor initiating cells, so-called BTIC, which gives rise basically to tumor. So the way this cell duplicates is always it goes to a cycle of self-renewal with the remaining of its stemness, and then give rise to lots of other different cells. And after, of course, primary brain tumor has lots of these BTIC cells. After surgery and standard therapy, usually those are the cells that are more resistant and they survive the therapy. And then, depending on microenvironment, they can invade, they can proliferate, they can give rise to angiogenesis. So it's mainly depending on the microenvironment how that BTIC will behave. What does it mean? Well, depending on the very local, for example, if we have cold outside, of course, what we have, but here in this room, our microenvironment is warm. So regardless it is cold outside, we don't have our coats, right? And we respond to our local microenvironmental signals. So that's what cells also do. So if the signal signals around interactions with extracellular matrix, some other factors that are released from their surrounding. Those signals will signal to BRIC to grow and proliferate and make those new blood vessels. Or if there are other signals in that very local microenvironment, like hypoxia, ionizing radiation, and so forth, other factors will give push to those cells to invasion. And like any normal cells, those tumor cells on their surface, they have lots of different receptors which will respond to those specific signals. A pair of ligand and receptor that I will talk more about today is a G protein coupled receptor. Ligand is bradykinin and receptor bradykinin receptor. So what is bradykinin? Bradykinin normally forms usually an interface with blood vessels. It's a 9 amino acid peptide. It's part of a kinin kallikrein system, and it's usually upregulated in conditions such as hypoxia, tissue damage, inflammation, all conditions associated with tumor growth as well. It is a G protein coupled receptor. We know of two receptors, bradykinin receptor 1, bradykinin receptor 2, and they are also involved in calcium mobilization, and we know that from our work on normal astrocytes. So this bradykinin, you can see in this movie, is So let me go back. You will see 3 cells that are circled. Please pay attention to those. You will see calcium oscillations and then cell moves. Calcium oscillations have been previously shown to have importance in migration of tumor cells. So after application of bradykinin onto glioma cells that were loaded with calcium dye, we imaged those cells and recorded oscillations, and after those oscillations, cells moved. So which of 2 receptors, as I mentioned, there are 2 receptors, bradykinin 1 and bradykinin 2 receptor, which of the receptor would be responsible for these oscillations and movement? Well, we showed that it's the bradykinin 2 receptor that is overexpressed on glioma cells, and also on PDX, which I will talk about a little bit later. What is really important for this story and interesting is that there is antagonist of bradykinin 2 receptor that is approved by FDA, it is approved here in Europe, it is approved in China for treatment of hereditary angioedema. So there is a drug already out there that has been tested, went through all toxicology, clinical trials and whatnot, and is approved by agency for use in clinic. It's called peptide icatibant, but it's so this is generic name, and this is the registered name is a registered drug as Firazyr. However, I will also call it HOE 140, and that is its sigma name because that same peptide, but of less quality and purity, you can buy in sigma and use for experiments. So there will be those 3 names interchanging. So I have showed that bradykinin 2 receptor expression is in many different cell lines, usually used for experimental work. What is more important, it also expressed in patient tissue biopsies, and it seems with the gray there is a higher expression, which is this red color. Blue color are nuclei. Also, it is usually expressed around blood vessels. Now it's labeled green, and blood vessels are red. If the cells, those cells, glioma cell lines, are put in concentration gradient of bradykinin and let in concentration gradient for some time, we have shown that bradykinin causes those cells to move towards the higher concentration. So from low concentration to higher concentration, unlike in control where they move in all directions, as well as we block the receptor with two different blockers, and they also move in all directions. Also, the velocity is significantly increased, as well as the distance those cells traveled in bradykinin concentration gradient. What is more relevant for patients and for this research is to put those cells in more in more context of tissue. So I perform a slice invasion in brain slices, where I put acute brain slice, and this was mouse slice. On top, I plated fluorescently labeled glioma cells and created bradykinin concentration gradient. And after certain amount of time, those slices were fixed, and I imaged them on confocal microscope. And as you can see here, in presence of bradykinin, there are lots of cells hugging and wrapping those blood vessels that are labeled red, and they penetrate relatively deep into the tissue. While if I blocked the receptor with or without bradykinin, most of the cells remained on the top of the slice, and they were not as much associated with blood vessels. So after analysis, I've seen a significant increase of percentage of cells that were wrapping blood vessels, as well as deeper penetration of the glioma cells in bradykinin concentration gradient comparing to control or when I block the receptor. The effects are really due to bradykinin signaling on bradykinin 2 receptor, because when inducible shRNA cells were created, there was decrease in protein expression, as well as a lack of response to calcium, less cells. There was a significant decrease in cell migration, as well as significant decrease in the percentage of cells that were attached to blood vessels. So I've shown you that bradykinin increases invasive migration of glioma through bradykinin signaling axis, and there are two main questions after that. What we are going to do with that? So the question is, what is the mechanism behind this process, and is there potential for therapy? So we put forward a hypothesis that this bradykinin increases intracellular calcium, and that intracellular calcium induces release of glucose through the process of release of glutamate through the process of exocytosis. Well, why is that important? Well, it is important because it has been shown that glioma cells release glutamate and that has been demonstrated in vitro, in vivo, and in situ from various groups. And by Harris-Sontheimer group, it has been shown that glioma cells express AMPA receptors, and they have autocrine glutamate signaling that promotes glioma invasion. Now, in his model, the source of glutamate is system XC, which is transporter that transports one glutamate outside the cell in exchange for cysteine inside the cells. However, when this transporter is blocked, there is about 50% of glutamate that is still released. So there must be other ways of glutamate to be released, and we propose that is part of that could be through exocytotic glutamate release. Also, it has been shown that this glutamate released by primary brain tumor induces epileptic activity, which is one of the side effects that many patients experience, and it's very debilitating for the patient. So if there is a way how to at least reduce that, that would be very beneficial for patients. We have done some experiments of glutamate release, and we use two different cell lines, one that does not express system XC and the other one that does. And as you can see, in both cases, there is a release of glutamate after bradykinin stimulation, which is blocked after application of that blocker that I was mentioning earlier. And that reduction is significant. We also, to confirm that it is exocytotic release, we repeated experiments, but this time we used the blocker for glutamate transporters, allosteric inhibitor, Rose Bengal, and after application of Rose Bengal, there was a reduction, significant reduction of glutamate release, regardless of expression of system XC. Here we moved into a different model, which is called PDX model, where glioma cells, unlike cell lines, which are propagate in dishes, here glioma cells are propagated in flank of nude mice, and that has been shown to be the best model which best replicates the actual or maintains characteristics of the tumor, because cell lines, they lose lots of characteristics during propagation. We further confirm by adding a cocktail of different blocking expression of different OMs that are expressed in glioma cells, which are proteins that are important for association of vesicles on the membrane. So when we remove that protein, there was also a significant reduction in glutamate release. So with this, we feel quite comfortable that this is one of the mechanism for the bradykinin, how it works on glioma cells. And so after rise of bradykinin along the blood vessels, that induces glutamate release, which is then also autocrine signal for cells to move along blood vessels. And bradykinin is exactly where it is generated close to blood vessels. So now the next question is, well, does that have any importance for the patients? Because we know that there are many, many targets that failed. And so in collaboration, Vlad in collaboration with some people at UFB, found that there are two proteins of group of proteins that are of interest for that bradykinin axis signaling, that there are two proteins that there is, if they are overexpressed, there is a reduction in patient survival. And those two proteins are bradykinin 2 receptor and VEM2, the exact two proteins that are important for the release of glutamate, exocytotic release. Also has been shown that there is increase in survival for the patients that have lower expression of bradykinin 2 receptor. However, not all patients have higher expression of bradykinin 2 receptor. There is a portion of patients, because as I said, those tumors are very different between people, among themselves they're very different. So it's really naive to think that we would find one protein that is really, you know, overexpressed for all patients. And that also only emphasized the need for personal medicine. Again, we went back to our tissue from patients, and indeed, in some of all the grade 4 tissues that we had, indeed there is a group of tissue from patients with the higher expression of bradykinin 2 receptor. So the next question is there potential for therapy? Well, one of the things that makes the therapy really difficult for glioma is a blood-brain barrier, right? Because it's so strict and doesn't allow any compound to cross blood-brain barrier and enter to brain. Well, I mentioned you, as glioma cells is crawling along blood vessels, it also disrupts that barrier. And also, there was this peptide, fluorescent peptide tamara icatibant that was created, and Manoj from Vlad's lab, he did these experiments, and he injected tumor cells in the brains of mice, let them grow for a couple of weeks, then injected tamara icatibant, killed the mice, and then look at the overlap between tumor cells labeled green and tamara icatibant, and you can see here there is a huge overlap. There is some tamara in peritumoral tissue, which is beneficial because usually there are cells residing which most likely were not surgically removed in patients. And then in contralateral side there was no labeling, so icatibant does pass tumor-brain barrier. And then finally we did some in vivo experiments with mice. We injected PDX cells. After 2 weeks of treatment, we started a therapy with retroorbital injection of icatibant, and then sacrifice mice when they started either losing lots of weight or having seizures, which is an indication that they're very close to their endpoint. Also, 2 weeks later, Manoj did seq mice and look into the changes in tumor volume and cell invasiveness. So he cut slices and then measured based on that fluorescence. He measures the size of the tumor, and based on thickness, he calculated the volume of tumor, which was with icatibant treated mice. He showed that there was a 58% reduction in tumor growth. And then we also did Kaplan-Meier plot where we were injecting that icatibant to mice, and we have shown that there is about 30% increase in survival with the mice that were treated with icatibant comparing to sham-treated mice. So the next question is, of course, would that be effective for human? And I can say that we are lucky now that we are in China. One contacted people in hospitals, and we are about to start clinical trials with the patients that will be treated with icatibant. And with that, hvala for your attention. And I'm ready to take your questions if we have time.
Thank you, Adriana. Any questions? I think, how do you take this peptide? Per rolls, injection, intrathecal?
Well, patients will take
Subdermal.
subdermal, but mice, we did intraorbital injections. Yeah.
Thank you. How many patients?
Well, we will see. We are hoping about 30 patients per year, but it depends, you know, how many patients.
Blind
study.
Thank you. Okay. Adriana, thank you.
Will the patients also be getting the usual treatment or just this?
Oh, we can do that only after patient get their standard treatment. This is proposed as adjuvant therapy. None of standard treatment would be reduced or changed or anything like that, because so far there is no therapy for inpatient, and this would hopefully be adjuvant therapy that would help patients. Yeah, but standard treatment should remain.
Thank you. Thank you, Adriana. So, I will do the impossible. I will bring us some time. And then I will just address one of the key questions, and that is the question related to glioblastoma. One of the key problems in, let's say, neuroscience and also neurobiology and neurology, because it's, as we have heard right at the beginning, it's one of the poorly treated sort of forms of cancer. Now, what is the highlight of this and other cancers, solid cancer, is heterogeneity that is not only morphological but also genetic. So if you look at the pathological samples of brains of patients that actually died of glioblastoma multiforme means that in fact the histology is so varied that it actually induces obviously the name. It's like the Aristotle would say, yes, this is the disease. The origin of this disease is in fact that it generates a genetic sort of modification. So most of the cancers in the body are somatic cancers. They develop, there is an evolution of cells within the body. Now, and the question is, can we treat these cancers by immunohybridomas? So what are immunohybridomas? Immunohybridomas are cells that actually are not present in the body. They are cells that are made in a laboratory, and they are made from two types of cells, from the tumor cell and from the immune cell. And the idea is that, in fact, we facilitate the recognition of the tumor cells by the immune system. Another feature that is not only heterogeneity, genetic and morphological of tumor cells, the other feature that is very prominent is that, in fact, for cancer to proliferate in our body, these cancer cells have to become invisible to the immune system. Okay? So mechanisms of how they attain that is, of course, there are multiple, but the idea would be to enhance the immune system. So when in the first lecture it was mentioned transplantation of organs, typically when we transplant organs, we have to suppress the immune system. Okay? Whereas in treating cancer, we have to enhance the immune system to recognize the antigens that have been developed during the evolution of cancer cells. Okay? So these are immunohybridomas. Now, the treatment of immunohybridomas, bring us a challenge. And the challenge is in the mindset of the Western society, where all the new treatments that go through the FDA, for example, Food and Drug Administration, or EMA, European Medicines Agency, is that in fact we have to target in a specific manner. So future medicines are precision medicines. Precision means either it is precisely targeting one molecule, one target in the cell or in the body, or it is completely personalized. It's made only for Janez, Mitska, and so forth. So, and the reason why we expect, and the regulatory bodies actually require specificity, either personalized or kind of targeted, is that in fact, the reason for that is Paul Ehrlich. Paul Ehrlich received the Nobel Prize in 1908 for immunology. Before that event, he proposed that, in treating a disease, we need to invent a molecule that is specific for the reason for that disease. For example, for a parasite, he was treating syphilis. And he coined that name specificity by the term magische Kugel in German, but magic bullet. Now, and this was presented at a lecture in London, and that has such an influence that we still expect that, in fact, new drugs are going to be very specific. Okay? And then he, in fact, delivered such a drug in 1909. Salvarsan, it is a molecule that was used for treating syphilis. Very successful. Now, the question is, can we use this concept for developing drugs for treating cancer? And as I already mentioned, there are multiple targets in a cancer cell, not only one. For a cancer cell to become successful, it has to go through the evolution of acquiring the right mutated genes to proliferate in the body. And here I would like to make an announcement. Go and read the book that is translated in Slovenia by Siddhartha Mukherjee. The title of the book is The Emperor of All Maladies, Biography of Cancer. It reads really well. It received the Pulitzer Prize. And if you start reading it, you can't stop. And you learn about the cancer a lot. Siddhartha Mukherjee also made a very important address at the Pulitzer Prize ceremony that was in 2013 at the already mentioned Sloan Kettering Institute by Ihan. And that is that he said, if you want to study, you have to be focusing, you have to become that problem that you are researching. And he would show an example of a scientist that was the best example of how to make big discovery. That's Gregor Mendel. Gregor Mendel, focused on studying peas, and then out of that study, that was many years, it took many years, the common theorem of genetics. Okay, so Siddhartha Mukherjee, and of course, in contrast to the magic bullet, we need to understand that the cancer cells are cells that in fact have multiple targets. Now, hence the challenge for treating cancer is that in fact we need to address all these targets. And of course the Nobel Prize in 2018, revolutionized the treating of cancer by introducing the fifth column of treating cancer. How do we treat cancer? Resection, chemotherapy, radiotherapy, hormonal therapy, and now immunotherapy. And this immunotherapy is based on so-called checkpoint inhibitors. So pharmacy produced molecules that inhibit the inhibition of immune cells by cancer cells. And these are single molecules. They're extremely specific because they are monoclonal antibodies. They have affinity of picomoles, but they only target one molecule, one target. Okay? So, and of course, the other problem is this molecule is one for all. So if you take these antibodies and treat various cancers, there are at least 600 diseases, and you'll see that it doesn't function in everyone, in every form of cancer, not even between patients that have the same kind of cancer. So the problem is with checkpoint inhibitors is that this is a single target pharmaceuticals, and then they have serious adverse events and are not suitable for all cancers. Okay? So cancer cells carry several mutations, I have said, and different among patients of the same cancer type. So a potential solution is that, in fact, we need to actually take rid of this one for all drugs, and of course, invent a way that would actually bring and address and target multiple sort of targets at the same time. And one way to do that is to use cells. As in the body, we use cells in the immune system to actually target, and we call this a pleiotropic manner of engaging multiple targets. Okay, so we have been really impressed some time ago by this concept, and of course this led us to develop this hybridoma cells. And hybridoma cells basically are fused tumor cells with immune cells that are called dendritic cells. And hence we actually bring the antigens from the tumor cells to a cell that actually presents this to the other cells. So dendritic cells are like macrophages. They become angry if you stimulate them with the LPS bacteria. And these dendritic cells have been discovered in 1973 by Ralph Steinman, who actually received the Nobel Prize in 2011, and he died a few days before he was nominated for the Nobel Prize. So these dendritic cells, in fact, teach other cell types what is the antigen, and of course, in these soldiers, lymphocytes, then kill the cells that carry these antigens. And that's the idea, is not actually engaging soldiers that kill the antigen-carrying cells, but in fact teaching lymphocytes what to kill. So anyway, this technology, I think, has been then developed, and then I will just go briefly through this. So this is basically the fusion procedure. So we have cells, these are electrodes. If you put them into an electromagnetic field of 2 megahertz, these cells form dipoles, and they stick to each other. When they stick, we electrocute them and electroporate the membranes, and then these cells actually fuse between themselves. So green and red cells actually sometimes become orange, and that is the fused cells. Now, what is important in this is not that, in fact, the cells only fuse, but also the organelles inside the cells, because antigen presentation from the theory of cell biology requires the fusion of lysosomes. Lysosomes are organelles that are very, very important. There was a Nobel Prize in 1955 for this. And of course, the technology we have here, in fact, we have here 2 nuclei, 2 yedro, and all these green and red lysosomes actually are fused, they are yellow. So this is important. So hybridoma cells are new cells that in fact have fused lysosomes. So basically antigens that are present in cells of tumor cells are then presented to the immune system when they are infused into the body. So clinical trial was carried out, and the indication was prostate cancer, because this was very easy to get access to the patients. This was done all in Ljubljana in Slovenia. And basically these patients actually survive. When they are diagnosed with a form of castration-resistant form, they survive maybe a year, maybe maximum 3 years. And when the idea came that we will carry out this treatment, the idea is to actually take the biopsy, have tumor cells, make dendritic cells from the blood, and then electrofuse them, and then generate these hybridomas, and these hybridomas are then injected back into the body subcutaneously, not intravenously. Okay? And of course, we were surprised to see that there was an effect, but not in 6 months. It took many, many tens of months to see an effect. So anyway, I'll just cut the story short to show you that, in fact, the placebo and the treated patients demonstrated a very strong effect, and that effect was related to the reduction of one type of cells that are natural killer cells, and the survival time was actually 60 months, not a year or 3 years, but in fact 5 years. So in any case, this is the important part here. This is the survival of mixed population of cancer patients, and this is the control. And in patients that were at the early stage of disease, even after 120 months of observation, more than 50% of patients were still alive. This is very, very encouraging. Okay, so it's an example of where we can actually use this treatment for attacking solid tumors in a totally personalized way. And of course, we address the targets in a multiple fashion. And we just started a new clinical trial that has been announced on the 21st of November for triple-negative breast cancer. So we think this technology would be useful for solid cancers and also for glioblastoma in the future. And this is the team, all from Slovenia. Franco Lugnani is basically in Kras area. Simon was the key junior doctor, supervised by Andrej Kmetec. Linka has driven all this project together with Sasha and Matejka, and of course the immunologist Alois, and of course Matjaž, and of course all the rest of the team. So the team worked really strongly. And this is my acknowledgement slide. If there are any questions, happy to answer. Thank you. Yeah. Alessandro.
A very small technical question.
Yes.
From the generation of the hybridomas, how do you select them?
They're all autologous. So the yield of hybridomas is about 5 to 10%. Okay, and they basically the number of cells that actually are actively teaching the other immune cells is very, very low because we could not see any dose dependency. So in our clinical trial, we had various yields of tumor cells for obvious reasons. And then we plotted the amount of cells as a function of survival time of patients, and we could see no correlation, meaning that, in fact, we need an extremely low concentration of hybridomas to elicit this teaching exercise to produce an immune response.
So you re-injected this population of 10%, let's say?
Yes. Well, in our case, it's very likely it's maybe 2 cells, maybe 3 cells, yes.
Yeah, but you select them based on fluorescence, right?
Well, no, we don't select them. This is a suspension, yeah.
Yeah, this will become critical for glioblastoma. So you don't re-inject the glioblastoma and make, first of all, you cannot do it subcutaneously because you're going to make metastatic cancers.
No, this we prevent metastatic cancer by irradiation. So all these tumor cells plus dendritic cells are heavily irradiated. Yes.
Glioblastoma, Robert, will have to be injected inside the cavity where you remove the tumor.
That can be done, yes.
Have you tried any hybridoma with glioma cells?
It's trivial. This is trivial. Every cell can fuse.
Can I have a question also regarding it's related actually to the questions which were raised. In glioblastoma, of course, in healthy brain, the permeability of the blood-brain barrier for antibodies is kind of limited. Of course, brain with glioblastoma is not exactly the healthy brain.
Mm-hmm.
Could you suggest maybe a strategy to increase the permeability of the blood-brain barrier, or do you consider that the BBB would be permeable for the antibodies?
Well, there are parts of the brain that are continuously permeable. It's not, the dogma that blood-brain barrier is basically a barrier. There's some parts in the base of the brain where actually antibodies can go across. An antibody is 12 nanometers in size. So it can go through. And of course, the drugs now for treatment of Alzheimer's disease are based on these antibodies, and they go into the brain. So area postrema, that's the reason. Yeah.
I do think, Robert, that it's best to inject it and fill the cavity with hybridoma.
Yeah.
Because it takes time. One thing you told me, it takes many, many months. Remind you, glioblastoma patients are dead in 14 months max with the standard of
They
care.
don't have months.
Yeah.
It needs days. So it needs
Yes.
to be injected inside with the gel.
And maybe, with
Well, it's just an. This is just a kind of a black box thinking, Yeah. So it's just an idea of
It's great idea. It just have to be a little bit adjusted from other tumors for specific
Okay. So thank you very much for the questions. I would like, in the interest of time, I'd like to invite our colleague from Trieste, from ICGB, Alessandro Marcello, please, and he will now switch to another pathology that is related to long COVID. So SARS-CoV-2 actually induced a very prominent problem, and that is, first of all, how this virus actually enters the brain and how it affects the brain in terms that we see this, well, in a number of patients, this long COVID. Okay, I guess you address this.
Thank you very much for this kind of invitation. I'm a virologist, and I must say that I'm here to learn from all you experts in gliobiology. I'm not an expert in gliobiology at all. We just got into this topic very recently. I'm from the International Center for Genetic Engineering and Biotechnology, which is an international organization with headquarters in Trieste, nearby here. Of course, our mandate is to provide excellent research for our member states, and through a series of programs, grants and fellowships, and Slovenia, of course, is one of our member states, contributes to the center as in fully entitled to get benefit from our activities. My group is really a virology group. I've been working on different aspects of virology of different viruses. We have a basic program of basic research in virology, mostly related to the interaction of the virus with the host cell. Of course, we have a program of applied diagnostics, surveillance and antiviral drug development, as well as we are interested in emerging viral diseases. Of course, we have a long-standing collaboration with Tatjana Avzic and Miroslav Petrovec here in Ljubljana. And recently I met Robert, and we were discussing about some of our work, and I'm trying to illustrate to you what we have been doing with SARS-CoV-2, which of course we have been all involved in research in SARS-CoV-2 during the pandemic, and so in the last 4 years. As a center, we have been working a lot in diagnostic surveillance, but actually what I'm going to talk to you today is really a basic aspect of SARS-CoV-2 infection. And this came early during the pandemic when we isolated the virus, and we started developing the tools to work with the virus, and then we asked a specific question related to virus infection together with some colleagues in Milan. And this relates to the effect of SARS-CoV-2 infection related to the induction of DNA damage response. As you know, among viruses, there are viruses that induce a DNA damage response. Mostly are DNA viruses that are involved in this kind of response. RNA viruses, for some reason, are much less studied in terms of induction of this response. However, it's becoming more and more important to understand this DNA damage response in relation to inflammation, and we know that most of the disease related to SARS-CoV-2 is related to inflammation. So just to summarize a little bit the response, which is quite complex at the molecular level. So you have a damage, which can be double strand or single strand. You have a recruitment of certain markers of the damage, like such as histone 2AX or RPA, that then trigger a repair complex, as well as a signaling that usually promotes cell cycle arrest. There are some outcomes of the DNA damage response. As I said, it's cell cycle arrest as well as inflammation, but also you can have, if you have a sustained DNA damage, you can have genome instability and cell death, as well as tumorigenesis and senescence. So these are all outcomes of double-strand DNA break. So we explored, first experiment that we did was to infect cells. So these are just HLH7 cells or CALO3 cells, or cell lines using for SARS-CoV-2 work or virology work in general, and look at the DNA double-strand response of these cells. And we immediately noticed that some of the markers were consistent. So, for example, this is DNA PK, which is phosphorylated so on the left you have the infection, minus or plus virus at different time points, and on the right you have the controls of irradiation or HU, so hydroxyurea treatment. So, for example, DNA PK is consistent as a signal for so for double-strand break repair, as well as this is you see that well. So ATM is also conserved in terms of phosphorylation, while, for example, ATR is not activated. So ATR, which is signaling this part of the pathway, is not activated. So again, so we see a response, so we see a DNA damage response, but it's not canonical, let's say. So it's a bit altered in terms of the response. In particular, we also observed that, for example, if you look at the CHEK1, which is a critical effector of the signal that controls several of the downstream effects of DNA double-strand breaks, you can see that there is no phosphorylation. Actually, notice here there is a little bit of downregulation of CHEK1 at later time points. So again, there is a DNA damage response following infection, but is not exactly canonical. This is also shown here. This is immunofluorescent, looking at CAP1 response, H2X, as well as RPA response, and this is basically formation of foci in the nuclei. Again, you see the response that is consistent. Most visually, most interesting, if you do this kind of comet assay, basically you take cells, and if you have a DNA damage, so if you have breaks into the genome, you have the formation of a tail of a comet. This is the tail here. And clearly infection-induced tail formation, meaning that there's actually physical break of the genome of the cell, which is quantitatively shown here. So again, we have a response. As I mentioned before, CHEK1, instead of not being activated, is actually progressively decreased following infection. So you can see again here, CHEK1, following infection, you have a decrease of the protein. And one of the downstream controlled proteins of CHEK1 is the ribonucleotide reductase subunit 2, so is also down-modulated. Again, you can see that is decreased in terms of activity of protein, both in HUA7, CALO3 cells, which is another model used for infection, and while the mRNA, so it's a post-translational mechanism, so RNA of CHEK1 remains stable while there is a decrease of the protein. So how this impacts the infection? So we postulated, we hypothesized this kind of molecular pathway. So you have infection, you have the NTP shortage because, as you know, RRM2 is part of the RRM complex that is involved in the conversion of NTP to the NTPs, which are involved in DNA synthesis. So we postulated that if we impact the levels of RRM2, the complex is not active and you should have an arrest in S phase. And we actually showed this by monitoring both the levels of the NTPs in the cells that are all reduced following infection, as well as looking at the cell cycle. And again, you can see cell cycle arrest in S phase that is typical of this kind of shortage. So we have an effect on the cell cycling and on DNA synthesis. Now, we can rescue this effect by supplementing the oxynucleosides. This is an intriguing experiment. Probably visually is better to show here. Again, you have the comet assay, induction of the double-strand breaks and the formation of the tail of the comet. And if you treat with the oxynucleosides, you see that this increase of comet of tails is again decreased. So there's a rescue of the phenotype, and is also shown, for example, for H2AX, which targets the signal, which is a signal of double-strand breaks. Again, you have phosphorylation with the virus, but when you induce with the oxynucleosides, you have the rescue of this phenotype. So it seems to be a pathway that is affecting the cell cycle of the cell and can be rescued by the oxynucleosides. But how does the virus induce this pathway? So infection, you have a number of viral proteins that are expressed during infection. So we took all the proteins, transfected the proteins in the cells, and look at the level of CHEK1 and RRM2. And we found that two proteins, which is NSP13 and ORF6 are actually able to downregulate these two proteins independent of the infection. So they are really targeting this pathway and inducing a degradation of CHEK1 and therefore ARM2, which is shown better here. So you have, this is CHEK1 levels and plus ORF6 or plus NSP13, they're both reduced. And how does it do that? Okay, I will not go into much of the detail here just for the sake of time. I'll just show you what we have shown before ORF6. So we have ORF6 is a protein that targets the nuclear pores and affects the nuclear cytoplasmic trafficking of protein and RNA, so it has different effects. So we believe it blocks CHEK1 in activity in the nucleus and promotes its degradation in the cytoplasm. In fact, you can rescue this effect by treating with MG132, which is an inhibitor of the proteasome. Interestingly, if you use a mutant of ORF6, this is a mutant M58R, this is a mutant that is not able to impact the nuclear pore, so is inactive, and then you can see that this effect is not there if you use the mutant. So we have shown that ORF6 is active in promoting CHEK1 downregulation into the infected cell, and this causes the double-strand break and the damage of the cellular DNA. And SP13 also causes CHEK1 reduction independently of 6 through the autophagy pathway. And then we have another mechanism that I will not talk today is about the nucleocapsid protein that impairs the recruitment of 53BP1 to the site of double-strand break, thus blocking the repair. Is this relevant also in vivo? So here we show mice as well as human samples. These are mice lungs, where we can see that there is accumulation of DNA damage also in the mice, and this recapitulates many of the markers that we have shown in the cells. You have reduction of C53BP1, activation of RPA and gamma H2X, and CHEK1 reduction. In humans also we could show these are lungs from COVID patients. Again, we see an increase of gamma H2X as a marker of double-strand break both in the lungs and in the nasal mucosa. A reduction, for example, in this case of CHEK1 both in the lungs and in the nasal mucosa. So this is an effect that we see both in vitro, so in cells that are infected, and in vivo. So one effect of having DNA breaks in the genome is the release of DNA in the cytoplasm, and this triggers a pathway, which is the cGAS-STING-dependent pathway, which activates inflammation. And in fact, we could see both a signature of inflammation of cells infected by SARS-CoV-2. This is the signal again of double-strand break, so release of DNA into the cytoplasm, inflammation, and most importantly, activation of cGAS. And these are markers that shows that cGAS is actually released in the cytoplasm associated with DNA, and these are other markers of cGAS activation. So we have activation of cGAS, we have an inflammatory response, and also we have an aging phenotype. So this is beta-galactosidase staining of cells following infection, both a cell line HUH7 cells that you can see that you have more signal in an infected cell. This is the control, which is the oxyurea. But most interesting, these are primarily nasal epithelial cells that again show the beta-gal signals in the infected cells. Why is this important? Well, I just want to conclude. This is a work that has been recently published, and all these conclusions that I made are described in this paper. And this has been a collaboration between our group, a group in Milan at IFOM that is an expert in double-strand break repair and genome instability, as well as other collaborators in Trieste and elsewhere. But as I was mentioned before, why is this important for the pathology of SARS-CoV-2 and for eventual therapy? This is because signals and stress and as infection induce a senescent phenotype, as I showed you before, the senescent phenotype characterized by DDR, so double-strand breaks, foci of gamma H2X, as we showed, the CGAS activation pathway, as well as other telomere shortening. We also showed the SCAP we still work on. So we have the senescent cell phenotype. This cell produces a so-called senescence-associated secretory pathway that induces senescent cell accumulation and chronic inflammation. And these are hallmarks of the pathology of SARS-CoV-2. We were interested to understand how this affects certain long-term features of the infection that can be summarized in general as long COVID. I'm not a clinician, so I won't engage too much into that. I'm just interested in the molecular pathways. So we needed a model to study infection of the brain by SARS-CoV-2. So we collaborated with the group of Michele Giuliano at SISSA in Trieste. They are more expert in neuropathology, and they had this model of ex vivo brain cortex of rat that we infected with SARS-CoV-2. So this is work that Pamela who is also here, has been much involved, also in the previous one. And the point at the beginning was, can we infect rat cortical cells? So the idea was that first we try to transduce AC2, a receptor, the human AC2, to get the cells infected. But Pamela came to me and said, well, actually the control gets infected very well, so apparently these rat cells are easily infectable also without the human receptor. Interestingly, if you infect these cells and you look at the cells that are being infected, you can see that mostly it's missing a graph. For some reason it's lost. So there was a histogram here showing basically that mostly glial cells are being infected with respect to neurons. So neurons appear not to be much infected, while astrocytes or glial cells are really the target of infection. Unfortunately, the graph is not shown. Not only that, not only we have infection of astrocytes, we have also an inflammatory signature here shown with an ELISA of different cytokines and chemokines. And this pathway is CGAS dependent, which is most important because it links to the previous work. So if you treat, so you can see, for example, activation of RANTES or activation of TNF-alpha, if you treat with a CGAS inhibitor, you have an inhibitor of this response. So it seems to be that that infection of brain cortical cells by SARS-CoV-2 induces an activation of a CGAS pathway that induces an inflammatory response. But what happens in terms of the damage response? So we took again the cells and we look at the different cell types. If you look here at astrocytes, you can see that this is the marker of astrocytes, and here are the foci characteristic of gamma H2AX histone, which is a marker of DNA damage. So we can see that astrocytes that are infected by SARS-CoV-2 actually induce a break into the genome of the cells, and probably this is the cause of the CGAS pathway activation and inflammatory response. This again shows H2AX, and this is important because we spend a lot of time trying to find the right control for these experiments, because we have SARS-CoV-2 infection, of course, we have mock, but what is the real control here? So we used UV SARS-CoV-2, UV-treated SARS-CoV-2. This is the virus treated with UV to inactivate the replication of the virus, so the virion is still present in the supernatant. It's the same preparation as the SARS-CoV-2 virus, but is completely unable to replicate. And this, to us, is the most important control also for what happens next, because next we will look at the effect of the infection on the activity of the neurons measured by micro electrode arrays. So basically you can see the cells. So this is a cerebral cortex removed from the brains of rats, and these were seeded on arrays of microelectrodes. So this is a chip where you can seed the cells, and then we can infect these cells with either with the SARS-CoV-2 fully active or with the control UV inactivated virus, and then monitor the electric activity of the neurons. And as you can see that there is, before infection, there is the normal electric activity, then this decreases following infection. And this is the quantitative data also showing, I don't see very well, but anyway, we could see that not only that you have an inhibition of the electric activity of the neurons, but also, and it's not shown here, I missed the slide, but we also could rescue this inhibitory activity by using a cGAS inhibitor. So cGAS inhibition, which is the trigger of the inflammation related to the infection, appears to be determining also this block of electric activity of the neurons. Of course, these are rats. Rats are not humans, so we try to, and here comes also my interest to interact with you about astrocytes, because it's not my field. CISA provided us some human iPSC astrocytes, and we could show that they can be infected. So you have these are plaques forming units, so it means that this is infectivity related to SARS-CoV-2 in these cells, and we different multiplicity of the infection. These are basic study looking at the infection of these cells, so you can see the signal also of the spike protein or the nucleocapsid in these cells, and we could also show markers of senescence and inflammation. In this case, KMH2AX is the usual marker of DNA double-strand breaks, but also you can see P21, which is a marker of senescence that is increasing in these cells. Also here the histogram does not appear, so I didn't check properly the slides when I loaded them here, and sorry for that. It happens sometimes when we transfer from the app to the Mac to the PC. So I will close here. Thank you for the attention. Here is I have to thank a lot of people and a lot of sponsors for our work, and particularly the work of Pamela, who has been a major contributor on this work. Thank you very much. Questions? Zala?
Okay, so first thank you for your very interesting presentation. You mentioned that SARS-CoV-2 mainly impacted astrocytes, not so much neurons, but did you observe some of the neurons that were infected?
Yes, there is a little portion of the neuron that is infected. There is also loss of neurons during following infection in terms of number of cells. But clearly, there's much more signal in astrocytes. Yeah, it's a good point, because you can also you have to look at also at the abundance of each cell types in the culture. But we checked for that, and I think there is much more infection of astrocytes, yes.
Can I just ask, excellent talk otherwise,
Thank you.
about the variant that you used. Did you use
Good point.
variant or?
No, this is the one that we isolated in March 2020. So it's the first, it's not the Wuhan, it's the D614 that was circulating here also in Slovenia at the early onset of the pandemic. It's a good question. This needs to be also shown with other variants, and also the impact of the different variants in terms of infection of upper respiratory tract and the brain is still questionable. So we need to work more on that, understand also from the pathological side on patients if really you have infection with the different variants and also of the brain. Yeah, good point.
If I may, we also did some infection with the Delta variant, with this bad cortical cell, and they were very permissive to infection.
Thank you.
Thank you, Alessandro.
Thank you very much.
another lecture on SARS-CoV-2, and this will be by Borut Furlani. Who's a PhD student, and he will explain which cell type is infected by this virus.
Thank you very much, Professor. So I'll do a comparison of infection of human astrocytes and human neurons, and also the super resolution insight into the infection of human astrocytes with SARS-CoV-2. So today we heard a lot. Already about COVID. COVID is of course caused by SARS-CoV-2 that originated in Wuhan, China in 2019 with detrimental consequences for human health and economy. Now, although the most presenting symptom was pneumonia in patients infected with COVID, there was also significant neurological symptomatics that is still poorly understood. There has also been a debate in scientific society for quite a while whether SARS-CoV-2 enters human brain, but now it was unequivocally shown that SARS-CoV-2 indeed is present in post-mortem human brain, especially of those who had some neurological symptoms during COVID. And this virus was present especially in astrocytes. Now we also heard a lot about astrocytes. I won't go into that. I will just make some points why astrocytes are important in neuroinfection in regard to COVID. So first is, of course, they are abundant, so they could represent they represent a reservoir for viral replication. Secondly, they also, of course, form where is this? This one. They form a blood-brain barrier together with endothelial cells and parasites, which is crucial for neuroprotection. They also have a suitable metabolism, and additionally, they also release a myriad of immunomodulatory molecules. So neuroinfection could also be, of course, through impaired astrocyte function. So now, given that SARS-CoV-2 is highly mutagenic, we know that we have a lot of variants, so it mutated quickly. We infected human astrocytes and neurons with several SARS-CoV-2 variants, and we also studied this infection in relation to the ketamine, which is a potent modulator of exo and endocytosis, and is also frequently used in mechanically ventilated and intubated patients with COVID in intensive care units. So I will divide this presentation into three parts. First will be comparison of infection of human neurons and astrocytes with SARS-CoV-2. We infected cultured human astrocytes and human neurons with several strains, or variants actually in this case. So because these labels are so complicated, as you can see here, I will just label the first one as SLO variant because it was the most prevalent variant in Slovenia in autumn of 2020. And I will call it SLO variant just for the sake of this presentation, okay? This was later replaced by English variant or Alpha variant in early mid-2021. And in our study, we of course also included the more recent Omicron variant. Now, at the cellular level, the infection was monitored using confocal microscopy, but also using structured illumination microscopy, which is type of super resolution microscopy. And at this point, I have to emphasize also that this study was done in collaboration with Institute of Microbiology and Immunology, where they infected cultured cells, and they also performed PCR experiments measuring concentration of viral RNA in cell culture supernatant, as well as the concentration of infectious virus. Okay, so here please observe fluorescent images obtained using confocal microscopy. On the left hand side, you can see infected human astrocytes, and here you can see human neurons. Now the nuclei are labeled in blue, and infected cells are labeled in green using antibody against viral RNA polymerase. And you can immediately see that there are much more infected human astrocytes compared to cultured human neurons. So even from these fluorescent microscopy images, we can see a higher rate of infection in case of human astrocytes versus human neurons. But to validate these findings, colleagues at Institute of Microbiology performed polymerase chain reaction in culture supernatant to measure the concentration of released viral RNA for 3 variants, as you can see here, for Omicron variant in this upper panel, for SLO variant, and for English variant. And you can clearly see that in this time span of 96 hours, the replication was much higher in astrocytes, whereas it was barely visible in neurons. Okay? But a crucial part of viral infectivity is also if the virus is infectious or not, because not all viral particles can infect other cells. So to this end, microbiologists use tissue culture infectious dose. I won't go into the details about that, but you can clearly see here that astrocytes produced infectious viral particles while neurons did not produce any. Okay? So to conclude part 1, I would say that human astrocytes much more efficiently, of course, produce infectious virus. So SARS-CoV-2 infects human astrocytes more efficiently than human neurons. And additionally, human astrocytes produce infectious viral particles, which together supports the role of astrocytes in SARS-CoV-2 neuroinfection. Now to move to the second part. The second part would be a super resolution insight into the SARS-CoV-2 infection of human astrocytes. So how is this infection visible with super resolution microscope? To repeat the timeline, PCR was performed every 24 hours, but additionally at 48 hours and 96 hours we performed immunocytochemistry for structured illumination microscopy, so this is type of super resolution microscopy. And we specifically targeted AC2, which is a receptor for SARS-CoV-2, and additionally two viral proteins, RDRP, RNA-dependent RNA polymerase components, and envelope protein. This way we could actually follow the life cycle of the virus, from the expression of the receptor to RDRP, which is integral to replication of apparatus of SARS-CoV-2, and then to envelope, which could be associated with maturation stages, and finally release, of course, which was monitored using PCR. Okay, and I think there are graphs missing here. Okay, this was also the problem before, that some graphs were missing. Huh. Let me see how it's along the way. Yeah, I will use words and show images, okay? I don't know why. But okay, nevertheless, the story will be here. Firstly, we validated well-known fact that human astrocytes express AC2, which is receptor for SARS-CoV-2, which makes them susceptible, of course, for SARS-CoV-2 infection. And interestingly, we also found that infection with SARS-CoV-2 upregulates AC2, so leads to the higher expression of AC2, as well as redistribution of viral particles, as you can see here, towards periphery. Periphery is marked here between these two lines. Okay, so this supports some differences in compartmentalization or redistribution of AC2 and its quantity upon infection. And a bit different results were found for different variants, which I cannot show because the graphs are not here, but anyway. So there were some differences between the variants. As I mentioned before, the rate of infection was monitored using confocal microscopy. Here in the upper row, you can see uninfected cells, and then in other 4 images, you can see infected cells. Infected cells are labeled in green. And we determined the percentage of infected cells at two time points, and the percentage was about 40 to 60%. So 40 to 60% of the astrocytes were infected with SARS-CoV-2. And additionally, the infection, of course, with SARS-CoV-2 led to gradual decrease in viability of astrocytes, which can be also visible here. In control cells, of course, you have more cells than here. So the infection led to apoptosis. So this is a super resolution image of SARS-CoV-2 infected human astrocytes. At periphery, you can see in red receptors for AC2, so they are preferentially located at the cell periphery. And viral proteins, RDRP, which is RNA-dependent RNA polymerase, is labeled in green and is located mainly in the cell center. Beside the nucleus, of course, in these replication organelles, where it also forms a slight indentation, as it can be here visible. So this was quantified using this modification, or nuclear deformation was quantified using circularity or shape factor. And here again, I don't know why the graph is not here, unfortunately. So, but the infection with SARS-CoV-2 led to a decrease in circularity. That means that the nuclei became actually deformed upon infection, which can, of course, influence a myriad of gene expression processes, as well as these mechanical changes could potentially inflict some DNA damages, as we heard before. Okay? The next protein is envelope protein, which is labeled here, and is mostly, of course, associated with maturation stages. Here with these two images, I want to emphasize what we can see, the differences in distribution of both proteins in infected astrocytes, where in envelope protein you can see much more homogeneous distribution as in comparison with RDRP, which is more centralized in, of course, replication complex. It's a trivial observation, but nevertheless I thought it's interesting. The last part is, of course, the release of newly synthesized virions, which is monitored by measuring concentration of viral RNA in supernatant, and both variants that were tested, alpha variant and SLO variant, replicated extensively in human astrocytes to similar extent, although we saw that there might be some differences at cellular level. So this concludes the super resolution insight into the SARS-CoV-2 infection of human astrocytes. Moving to the next part, which is a possible role of ketamine in SARS-CoV-2 infection. So ketamine is a sedative, anesthetic, and also a potent antidepressant used in some severe cases of depression where other options are not viable. And it's also frequently used in intensive care units in patients with severe disease that have to be intubated and mechanically ventilated. Some clinical studies already actually exposed that ketamine could have some anti-inflammatory effects after, for example, some surgical procedures. The inflammatory markers were reduced, and so on. And also at the cellular level, colleagues in our laboratory showed that ketamine influences exocytosis and endocytotic processes, having inhibitory effects, which led us to a question if ketamine has possible also implications via this mechanism on viral entry or release. Okay? So additionally, some of the samples were treated also with ketamine, and we observed, in case of the SLO variant, that ketamine treatment actually resulted in a reduction of viral RNA synthesis at later stages of infection, but this was not pronounced in case of the alpha variant, nor it was pronounced in the case of infectious viral particle production. But nevertheless, utilizing super resolution microscopy, we could see that ketamine treatment reduced RDRP, so RNA-dependent RNA polymerase, particle density at early stages in case of a SLO variant, but also in the case of English variant at later stages of infection, at 96 hours post-infection. So this means that ketamine might possess some strain-dependent effects on SARS-CoV-2 infection of human astrocytes, either via reducing RNA synthesis, so in these replication organelles, or by some inhibitory effects on exocytotic mechanisms via, for example, cyclic AMP. So this would conclude my presentation. My take-home messages would be that SARS-CoV-2 much more efficiently, of course, infects human astrocytes, the neurons, and only human astrocytes produce infectious viral particles, as well as there are some differences between the strains, especially at the cellular level, and also lastly, depending on SARS-CoV-2 variant, ketamine might possess some beneficial effects on infection. So I would like to thank my supervisor, Professor Jurgachevsky, and of course Professor Zorec and other members of our lab, as well as colleagues from Institute of Microbiology and Immunology that made, of course, this study possible by infecting cells with SARS-CoV-2 strains and then measuring other molecular things. Okay, so thank you for your attention, and now.
Well, thank you very much for infecting us. There are questions there.
I have a question. Thank you for that very nice presentation. Could you maybe also quantify some morphological aspects of these infected astrocytes?
We did not, but yes, it's a very nice suggestion, like circularity, for example, of the cells, which probably, of course, was reduced because the cells became, I think, more, yes, less circular, actually, or, yeah, thanks. Yeah.
I have many questions that I will ask you later, but
yeah.
One is about the image on the infection, that you see this band of the nucleus and this real circular
This one?
RDRP localization, which
Yeah.
is very peculiar. It reminds me of Zika virus for some reason. Because I think I observed the same thing, the bending of the nucleus in Zika. I wonder if you have ever checked that as well.
In case of Zika virus? In case of Zika virus, this was we did not check. Irina Jurgachevsky says this was done before my time, before I came to the laboratory. But I think that all the images were performed using classical epifluorescence. Maybe it was not visible there. This is structured illumination microscopy. It's better, of course, to distinguish such structures. But Irina Jurgachevsky, I think it's not. You did not observe.
I do not think it's not. We unfortunately at that time did not use the puppy labeling, so.
You did not. That's why. Mm-hmm.
Just a curiosity.
Yeah.
So I guess you can have questions over lunch, and now we go.
Thanks.
Now we have come to our last presentation that will be online. I guess we have Professor Dmitry Krainz in Chicago. I guess he's with us. And I will ask Rok at the back to actually to link us. So Professor Krainz will talk about the role of mitochondria lysosome contacts in health and disease. I should mention that Professor Krainz is a very prominent neurologist in the United States, originating from Celje, and then did his training in this area. But now he is also the president of the Neurological Association in the United States, and also a very good friend that actually would like to extend his activities over down to Slovenia and of course the region. I guess he can hear me and then
Yeah, I can hear you. Thank you. Can you see me and hear me? Can you hear me?
Yes, we can hear you.
Good. Well, thank you so much for inviting me. I'm sorry I'm not there in person. You know, I love coming to Slovenia, but I was just there recently and I couldn't get time off to come back. But I've been listening to your talks. It's very early here in US, but I have listened for the last few talks. Really, I see a little bit of an echo. I don't know, maybe we should mute something. I don't know what to do, but can you hear me now? Okay, so I assume you can hear me, although I cannot hear you, but I want to talk about mitochondrial lysosomal contacts that we discovered recently. And it's actually an exciting new area, these organelle contacts, and I'd like to spend a few minutes on that, but I want to tell you how we got to this. So we were, first of all, I think some of this was mentioned today, especially in neurons, mitochondria and lysosomes have been implicated in a number of different neurodegenerative diseases, especially Parkinson's and also ALS and Alzheimer's. And we know the dynamics of these organelles is really important in neurons because they have to travel up and down axons into synaptic terminals and provide energy at the synaptic terminal. And we've been studying these organelles in mostly in neurons, but I will mention a few things that we're trying to do in glia as well. And one of the major projects that we've had over the years is trying to understand how these two organelles communicate in a functional way in Parkinson's disease patient-derived neurons. So this is a patient-derived through iPSC. So we take skin biopsy, we make induced pluripotent stem cells, and then we differentiate those cells into dopaminergic neurons. And because there are so many different genes linked to Parkinson's disease, over 200 genes, we wanted to understand how those genes, and most of those genes are linked to lysosomal function or mitochondrial function. We wanted to understand how they converge to cause degeneration of these dopaminergic neurons. And that's the work I'm not going to talk about today. But during this project, we made an unexpected observation that actually, in addition to the functional connection between two organelles, there was a direct contact between mitochondria and lysosomes that really caught our attention. And then we try to pursue this in more detail. So this is like a video I can show you. You know, lysosome is green, mitochondria is red. And you can see how lysosome is sort of attached to mitochondria. And these contacts, they last less than 1 minute. And we used different technologies, EM and super resolution microscopy and confocal and FRED imaging to really understand the nature of these contacts. And we actually showed that it's not related to any degradation. As you know, mitophagy has been well described, where lysosomes take up dysfunctional mitochondria and degrade dysfunctional mitochondria. So we show that this is not related to mitophagy. This is a whole different process. And what I'm going to tell you today is how these organelles communicate in neurons, and we're also trying to do this in glia. And basically, they are exchanging metabolites during these contacts, and I'm going to show you a few experiments about that. So which proteins regulate these contacts? That's really important to understand the molecular machinery that's responsible for these contact formation. Because if you don't know the machinery, it's really hard to study the contacts. First, you think, maybe this is just a random contact between organelles in a cell. But if you really know the machinery, then you can manipulate the contacts and study them in more detail. So we have looked at this, and I don't want to go through details here because it's published, but just to mention that the main proteins involved in these contacts are GTP bound RAP7, which mediates contact formation. And then FIS1 protein that sits on mitochondria recruits TBC1D15 gap activity to hydrolyze RAP7 GTP, and that untethers the contacts. So there's a tethering and untethering component of these two organelles, how they come together and how they go apart. And we have dissected this mechanism in some detail, but I'm not going to talk about this too much, except to say that to study the function of these contacts, we use this machinery as a tool to really understand what is going on with these contacts. So one thing I'm going to mention is regulation of mitochondrial dynamics through these contacts. One thing we noticed is that mitochondrial fission is preferentially marked by lysosomes. So when lysosomes connect with mitochondria, they promote fission of mitochondria. And that's not the case when we looked at peroxisomes or early endosomes. It's really lysosomal mediated effect. We have also noticed that during these experiments that mitochondria, they form contacts with each other. The traditional view is that when mitochondria come together, they fuse. So fusion of mitochondria has been described pretty well. But what we noticed is that fusion is actually a minor event. When you compare the frequency of mitochondrial fusion with the frequency of tethering and untethering mitochondria, I can show you the quantification here. So most of the time when mitochondria come together, most of the time they untether. And in very few cases, they fuse together. So that's quantified here. One of the consequences of this tethering, which again is with less than 1 minute, all these tethering events were in general less than 1 minute. There was a very small proportion of organelles that were tethering for more than 1 minute. And these intermitochondrial contacts then restrict motility of mitochondria. That's one thing that we noticed in cells. Now, lysosomes, when they touch mitochondria, when they form these contacts, they actually promote untethering of mitochondria. So that's one of the consequences of these lysosomal mitochondrial contacts, that lysosomes promote untethering. And we described the mechanism in detail, but I'm not going to go into that today. So anyway, what I showed you so far very briefly is that mitochondrial lysosomes, they form contacts through this machinery, and they promote mitochondrial fission. And also lysosomes promote untethering of mitochondrial contacts, mito-mito contacts. We call them mito-mito contacts versus lyso-mito contacts. And we have a mechanistic insight into this, but I don't have time today to go over this, but I just wanted to alert you that the two processes are coordinated through outer mitochondrial membrane proteins, FIS1 and MID51. We show that these two proteins form oligomers on mitochondrial membrane, and they attract TBC1D15 and DRP1, and the DRP1 GTP hydrolysis and RAP7 GTP hydrolysis are coordinated in these processes. And we described this in our Journal of Cell Biology paper last year. So it is a highly regulated process. It's not random by any means, but it's regulated. We're still trying to understand what is the signaling pathway that regulates these processes. Okay, so what is the function of these contacts in disease, and are they disrupted in disease? This is sort of the bulk of my talk today, but I just wanted to set the stage by showing you this brief introduction. Now, when you think about disease, you know, it's an unlimited number of options, right? If you think about lysosomal mitochondrial contact formation is a very fundamental cell biological observation. And you can study this pretty much in any disease. But our choice of diseases to study was based on genetic mutations that are linked to mitochondria or lysosomes. This was a natural initial step. So, you know, the assumption being if there's mutation in lysosomal genes, that will have some effect on contacts because lysosomes will be abnormal. If there's a mutation in mitochondrial genes, we should see some problem with contacts because mitochondria will be abnormal. So we looked at the mutations that are linked, that are listed on this slide. One interesting disease to look at is Charcot-Marie tooth, which is a type of neuropathy, because the genes that are mutated in Charcot-Marie tooth are overlapping with the genes that we identified, or proteins that we identified that are involved in tethering of mitochondria with lysosomes. So that's the first one we looked at. And then we also looked at TRPML1, which is calcium efflux. When TRPML1 is mutated, you get mucolipidosis type 4. We looked at GBA, which is a lysosomal enzyme linked to Parkinson's disease, and Parkin that's involved in mitophagy. So I'm not going to go through all of these, but I'm just going to mention what happens with mitochondrial lysosomal contacts when you have mutations in lysosomal or mitochondrial genes. Charcot-Marie tooth is the one I mentioned. That's where we saw significant abnormalities in mitochondrial lysosomal contacts because the genes that are mutated in this disease are the same genes that are linked to regulation of contact. So this is maybe the most striking phenotype that we saw. It's published. I'm not going to go through this in detail. I want to tell you more about the other two examples. So one example is in Parkinson's disease. We looked at GBA mutation. GBA is a lysosomal enzyme that is the highest genetic risk factor for Parkinson's disease. It's normally involved in lysosomal lipid metabolism. And when it's mutated, lysosomes are dysfunctional, and that contributes to degeneration of dopamine neurons in Parkinson's disease. And degeneration of those neurons is responsible for all the main symptoms of Parkinson's disease. So we asked a simple question, you know, when lysosomes are dysfunctional, what happens to contacts between lysosomes and mitochondria? And this was a project by my graduate student. And on the left, you see that we have lower GCase. GCase is the protein coded by GBA. GBA is the gene, GCase is the protein. We have lower protein expression, we have lower activity of this enzyme. And so this is in human neurons. I forgot to mention, this is patient-derived neurons. So we take skin biopsy from patients who have GBA Parkinson's disease. We make induced pluripotent stem cells, and then we reprogram those cells into dopaminergic neurons. So we have shown that in those dopaminergic neurons, the contacts between mitochondria and lysosomes, they form in different parts of neuron, in soma, in axons, and in dendrites. Now, what Sujin Kim, who was my grad student, what she showed in these experiments is that the contact duration was increased in these patients. And she spent a lot of time doing controls for this experiment because the assumption was is it a non-specific effect of lysosomal dysfunction? So when lysosomes are dysfunctional, they're not as mobile. Maybe that's why they have more contacts. So she has shown that that's not the case. This is a more specific mechanism that involves one of the tethering factors, TBCD15, and the mechanistic experiments are described in the paper. I just want to mention that the contacts were increased, and one of the consequences of the increased contact was that lysosomal mitochondrial distribution to synaptic terminals was decreased. They were sort of hijacked by lysosomes. And one of the consequences of that was that there was less ATP available at the synaptic terminals, which led to synaptic dysfunction in these patient-derived neurons. And Sujin was able to show that expression of TBC1D15 was able to restore some of these pathological phenotypes. Again, this is published. I don't have to go into details here. So in the summary, what I told you about GBA1PD is that we found prolonged duration of mitochondrial lysosomal contacts in human patient-derived neurons, and this resulted in disrupted function and axonal distribution of mitochondria. And we were able to partially normalize contacts and then restore mitochondrial function in GBA neurons. I mentioned before, there are over 200 genes linked to Parkinson's disease. This is just one gene. So we wanted to test at least another example of genetic Parkinson's disease. And we decided to go with this gene, which is it's called Parkin. Homozygous loss of Parkin causes autosomal recessive Parkinson's disease. The reason why we chose Parkin is twofold. Number one, Parkin, as you know, you probably know pink Parkin connection in mitophagy. So pink activates Parkin, and then Parkin promotes mitophagy, which is degradation of dysfunctional mitochondria by lysosomes. This is a well-described pathway that was discovered by a few different labs not that long ago, and a really important quality control mechanism in a cell. The other reason why we chose Parkin is that we have shown in the past that Parkin activates RAP7, and I told you that RAP7 is an important tethering factor in mitochondrial lysosomal contacts. And so we assume that something's going to be wrong with the tethering, because if Parkin is mutated, RAP7 function will be abnormal. We have also just published a paper listed here in Neuron where we looked at Parkin in the regulation of synaptic vesicle endocytosis. So this was another reason why we focused on Parkin. And what we found with Parkin was that, and this was my MD PhD student, Wes Peng, the paper just came out this year. He showed that the contact duration is decreased. And this was not terribly surprising, because maybe that's my next slide, because he was able to show that RAP7 activation was decreased by mutation of Parkin. And again, we're talking about human patient-derived neurons that we generated through iPSCs. And in those neurons, Parkin activation, mutant Parkin, does not activate RAP7 in the same way as normal Parkin. So I think that's the reason why, and here we used Parkin that has ubiquitin ligase mutation, and that Parkin is inactive. So it's ubiquitin-related effect, as you can show in the pull-down experiment. So that's the main reason why we think that the tethering of mitochondria and lysosomes was decreased in Parkin. Now, what is really interesting, and this is what I have to say, this is what I'm most excited about, is the experiments that were done as part of this project, where Wes was able to show that when you purify mitochondria and purify lysosomes, and you do metabolomic study, and then you modulate the contacts between mitochondria, you make them more or less frequent. He was able to show that there's an amino acid transfer from lysosomes to mitochondria. Not all amino acids, but some amino acids are being transferred from lysosomes to mitochondria through contact. It's a contact-dependent effect. And this is what I mentioned before. If you know the machinery that regulates context, then you can really understand what is context dependent, what is not context dependent. Now, it's not super surprising that lysosomes that normally degrade proteins and they produce amino acids as breakdown products, that they would supply lysosomes, mitochondria with amino acids, because mitochondria, they don't have amino acids and they have to import them. And the mechanism how mitochondria get amino acids wasn't that clear. So this is sort of a new concept in terms of how mitochondria get amino acids from lysosomes. So we are really working on this now, trying to understand what else is being exchanged between the two organelles. This is the summary of the mechanistic details that I'm going to skip this for the sake of time because I only have 20 minutes, but it is published in our recent paper. We have some understanding how these amino acids are transferred, what are the key players in this transfer. But the bottom line is it is a context-dependent transfer of amino acids. Naturally, we wondered what else is being exchanged between these two organelles. Now, remember, they come together for less than 1 minute, right? This is a short contact that you can only see with live cell imaging. They come together and they come apart. So we assume that other things are being transferred during that triazine interaction. And one thing we looked at, we are looking at lipids and other metabolites right now, but we haven't published that. I don't have the data, but one thing that we did publish is the transfer of calcium from lysosomes to mitochondria. So for this, we used TRPML1, which is mutation linked to mucolipidosis type 4, which is a completely different disease. But the point being, when you have mutation of this gene, calcium efflux from lysosomes is diminished. And Wes Pang has shown that this calcium transfer from lysosomes to mitochondria is diminished in the context of this mutation. First of all, he has shown that there is calcium transfer. I can show you the video. So here's the video. Red is mitochondria, green is lysosome. You can see how the redness goes up and how the green goes down when this is measuring calcium, right? So calcium is being transferred from the green to the red. And Wes was able to show that this effect is mediated by TRPML1 channel because he had to activate TRPML1 channel in a normal situation to get calcium transfer. And then when TRPML1 channel is defective, then there was diminished transfer of calcium in disease context. And VDEC1 and MCU and mitochondria play a role in regulating this transfer. So that's so basically what I told you. There are two things that we know for sure happen when mitochondria and lysosomes come together. One is transfer of amino acids, and the other one is transfer of calcium. And we're currently working on lipids and ATP, which is a technical challenge. We're trying to figure out if ATP is moved from mitochondria to lysosomes. So that's kind of the summary, a quick summary of what I told you so far is it's a new pathway of understanding mitochondria and lysosomal crosstalk. The proteins, I told you about the proteins that regulate the tethering and untethering, and what is the function of these contacts in mitochondrial fission and fusion, and how these contacts are disrupted in some diseases that I mentioned. Now, we are currently working on human microglia and human astrocytes. So we generate, this is unpublished, but we are generating from iPSCs, so from the same patient or the same normal subject, that we can generate neurons. We can also make microglia or astrocytes. And we've been trying to understand what's the nature of these mitochondrial lysosomal contacts in glia. Again, looking at patients who have mutations in these organelles. And how does that affect microglial function, for example? So this is one example that you see here is microglia from patients, and the red is pH-sensitive myelin. So we can measure how much myelin is taken up by lysosomes in these microglial cells. And then we can use alpha-synuclein, we can use other triggers that are taken up by microglia. And if you have dysfunctional lysosomes, microglia become angry, so to speak, and that results on hyperactivation of microglia. And we are currently looking at all those aspects, but also trying to understand how microglia and the mitochondrial contacts, lysosomal mitochondrial contacts are regulated in glia when glia is normal or dysfunctional. And we're also making astrocytes from iPSC-derived cells and doing the same thing in astrocytes. Now, the ultimate goal of all this is to understand how glial dysfunction affects neuronal function. So for that, we are culturing, I have to move this, but I can show you this. So this is a co-culture of microglia and neurons. So these are patient-derived microglia, patient-derived neurons, and we can co-culture them and really study them together. That's the ultimate goal of this project. We've been spending quite a bit of time on understanding microglial function and dysfunction in disease, and then what is the effect of that on human neurons, dopaminergic neurons. Contacts are part of that story, but not the only part. So, but I just wanted to mention this because this symposium is about glia, and we have a lot of unpublished data on glia, and I would be happy to obviously collaborate if there's some potential there. Now, to study glia and contacts in glia, we have developed this, this is probably my last slide, but we developed this Robert Kukos, who's my recent postdoc. He trained with Alice Ting at Stanford for his PhD, and he's very technical. He learned a lot of interesting techniques with Alice Ting, and he's applying some of those techniques to study contacts between mitochondria and lysosomes. So, for example, he generated this technology where he can induce transiently, he can induce all the mitochondria to come into contact with all the lysosomes. And he can regulate how long to keep those contacts together. And then by removing abscisic acid, which is the trigger, he can, the contacts come apart. So I can show you this here. So you see green is lysosomes and red is mitochondria. So right now they're all separated, or most of them are separated. And when he adds abscisic acid, you will see that they all come together and everything becomes yellow. And when he removes it, they come apart. So this experimental system is allowing us to really understand the function better and to study the function of these contacts in a little more detail. So that's all unpublished, but we're trying to develop some of these technologies to advance these studies in glia as well. And that's the end of my talk. These are the key people that did the work that I just described. Yvette was a postdoc, very talented. She now has her own lab. Dan was a postdoc. He's now running a big group in a biotech company. Sujin works at Genentech. She was a graduate student in West Bank. Is a resident at Harvard in neurology. Robert is my postdoc, and Leon is my student. And this is our lab in Chicago. These are the people, this is at my house, a Christmas party. The guys who have stars above their heads are the people that I mentioned. This is Yvette, Dan, Wes, Sujin. Those are the key people working on this project. Everybody else works in my lab except these two girls here who are my daughters who sneaked in during the party. Thank you so much. I'll be happy to take questions.
Thank you very much. And we need to figure out how this will work. If there are questions, can you hear that? Dimitri,
I can hear you, but I cannot hear questions.
Well, we'll have to kind of improvise. Yeah. Yeah. Okay, so any questions? It's lunch break actually, Dimitri. So we will have to actually wait for the questions. And I'm sorry you cannot join us for lunch. We will do that next time you come to Slovenia. But absolutely, the questions would be: so what is preventing these contacts to go into mitophagy? That is my question.
Yeah. So no, that's exactly that. You know, when we had this paper in review in Nature, that was one of the questions by reviewers, right? So we have shown, it's in the paper, we have shown that mitophagy is a whole different process. Mitophagy happens when mitochondria are severely dysfunctional. We are talking here about normal mitochondria that are not dysfunctional, and the contacts that form in a very transient way, right? So they come together and they go apart after less than 1 minute. Whereas in mitophagy, you have the engulfment into autophagosomes, and then autophagosomes fuse with lysosomes, and mitochondria end up in the lysosome. And there are other people who have shown that, like Heidi McBride from McGill, that mitochondrial vesicles are detached from mitochondria and they're degraded by lysosomes. So we looked at that as well. So this was a very different process from mitophagy because we're really dealing with, for the most part, with normal mitochondria.
Thank you for the answer, and I'm sure there will be questions, and there is potential for collaboration, and I'm sure you will receive emails, and that, I guess, will be an idea to continue.
That's great. That would be wonderful. Please share my email with everybody.
Question.
Hello, very nice talk. I was wondering about the cytoskeleton. Have you checked these processes? How, to what extent, of course, they are dependent also on different types of cytoskeleton? For instance, microtubules.
Yevnei is asking whether microtubules are involved in this lysosome to mitochondrial contacts. Okay. Yeah. The question was whether Yernei was asking whether microtubules are part of these contacts between lysosomes and mitochondria.
That's an excellent question. Thank you for that question. Yes, we are. We're collaborating with people who are studying microtubules to understand whether or not this is regulated in terms of microtubule function. That's a wonderful question. We initially didn't study that, but I suspect, based on some of the early data, that yes, there's an involvement of microtubules. And related to that question, right? We are trying to understand when does mitochondria tell lysosome, hey, I need some amino acids, I need some calcium, please come close to me, right? What is the signaling pathway that regulates these contacts? That's of great interest to us. and how do they inform each other that they have to partner and exchange metabolites? And what is the role of microtubules as well in terms of their regulation? Yeah, these are wonderful questions.
Okay, thank you. And once an applause to you. Thanks, Dimitri, for your presentation. There were a little bit hiccups with the technology, but we managed. It's magic that, in fact, we can communicate. And right at the end, I'd like to express my sincere thanks to the team that actually worked out, especially Rok Letonia and Daria Rogel from the Academy. So we need to clap. And then we would also like to thank, of course, Nina Vardian, Annemarie Horvat, and whoever helped the Slovenian Physiological Society to put this meeting together. And finally, I would like to thank all the speakers coming from far away, from Trieste, but also as far as from China. And we hope that actually you have learned something today. And of course, we can have a discussion with the Culinary Delights at the Prešernova dvorana and Zupančičeva dvorana across the square, Novi Trg 3. Thank you.